Good afternoon, everyone. My name is Nick Reynolds, and on behalf of the IEC and IPC, I'd like to welcome you to this session. So I'm co-chairing this with Johan Gudjonsson, and I'll introduce him briefly in a second. In order to maximize the amount of talks that go on, we're going to give very brief intros to the speakers, but their bios are all available online through this QR code or through the system if you go through the regular ESDR app. And we've tried to bring together, we've done an IPC, IEC meeting before, which was very successful, and we think, given the development in science and translation across these two important diseases, that it was very timely to have another session to consider comparative pathogenesis and biomarker-guided treatments, which we're all aiming to do in the clinic, but always just seems out of the clinical realm. So I'm the education co-chair of the IEC. I don't know where, oh, here we go. Yeah, so the other thing, just a few housekeeping points. So this session is being recorded, and it's very valuable to have the resource available online. And the questions you ask will also be recorded, so please bear that in mind. And you can also send further comments or questions through these email addresses. So in fact, the IEC has been going now for 10 years and is rapidly growing. It now covers 32 countries. It's got 145 counsellors, and it's increasing its educational activity with increasing reach globally and covering all continents. Following this meeting, there's an important final session of this year at the EADV on hand eczema, which I encourage you to attend. So we've developed some mission statements and vision in line with many organizations. I'm not going to dwell on these, but I think it is important to highlight that a key aim is really putting patients at the center to improve patients' care by promoting research, education, and best practices. And we do have a very worldwide perspective. And we're really interested in transforming people's lives across the world. And our global reach is crucial in that line, and we're trying to bring together leaders from different continents to interact and encourage early career researchers to come forward and present at these conferences as well. So our values are patient-centered. Clearly, we're interested in innovation, evidence-based medicine, and being globally inclusive. We'd like to thank our sponsors who have supported us over the years, and also encourage you to connect on social media and promote this interaction between the psoriasis and eczema communities. So my final task is to introduce Johan, who is Professor of Dermatology and also Professor of Molecular Skin Immunology in Ann Arbor and has expertise in both fields, so he's perfectly placed to be part of this consortium. Thank you.
Thank you, Nick. Well, thank you, Nick, for that introduction. I'm here on behalf of the International Psoriasis Council. I'm on their board of directors. And I'm just going to go over a brief welcome from the IPC. We're very excited to be with you today. This is actually the first session that we've ever done or partnered with the IEC, and I hope we have an informative science to kind of show you today. I want to thank the IPC program supporter, Johnson & Johnson Innovative Medicine, for supporting this session today. And just to kind of give you a brief overview of the mission and vision of the IPC. So we're a passionate community of researchers and physicians experts who share a common goal, which is to improve the health and well-being of people with psoriasis worldwide. And we understand the challenges that people face with psoriasis, and we are committed to making a difference in the life and the management of these patients. So our vision is to create a world free of the burden of psoriasis, where people with psoriasis worldwide have access to the best available care and are empowered to lead healthy, fulfilling lives. So we are a global network. We have about 200 professional psoriasis experts or counselors that volunteer and represent over 48 countries. And then our global network has about 2,000 psoriasis enthusiasts from over 95 countries. And you can see the geographical distribution on the map on the right. The strategic areas of focus of the IPC are multiple. So one of them is to educate medical professionals through expanded outreach. It's also to strengthen the capacity of researchers to promote collaborations, expand and diversify our global network, improve the quality of care and access to medicines for psoriasis, and deliver a program based on regional needs. So just to kind of highlight, we have, in the coming year, we have at least 7 upcoming events that the IPC is sponsoring across the world at different major dermatological meetings. And I have the QR code there that if you want to get more information about these events, so please utilize that. And then also to stay up to date with the IPC, another QR code to kind of subscribe to our various ways that we distribute information, whether it's Instagram, the Twitter or X or Facebook. So with that, thank you for listening, and I think we'll move on to the first talk, which is actually the one that I'm going to be giving. And I got the honor of being the first speaker of the of today, and to kind of briefly introduce some of the things that we're going to be discussing this afternoon. The title of my talk is The Comparative Pathogenesis of Psoriasis and Atopic Dermatitis. It's hard to give this justice in just 15 minutes or so, but I'll do my best. These are my conflicts of interest. And then, I find it to kind of be very helpful to kind of just put up a photograph of psoriasis and atopic dermatitis. And in some ways, you can see how different these diseases are, but at the same time, you can also see how similar they are. They can be pretty extensive. The skin is red and inflamed, but you can also see, these subtle kind of differences between the diseases, the thick kind of silvery scale of psoriasis, and then the more kind of wet-like appearance of atopic dermatitis with sometimes marks of excoriations given the intense pruritus that is associated with this disease. I know the group here is very familiar with psoriasis, but it's always good and helpful to kind of revisit some of the features of psoriasis. So there on the right, you have some clinical photographs that I have taken over the years of cases that I've seen in the clinic, and just gives you an idea how variable this disease actually can be. It's a common disease. It affects about 2% of the US and European population. It's a disease that is genetically predisposed but complex, and that means that there are multiple different variants that contribute to disease susceptibility. And to date, we have identified about 160 of those. The age of onset of psoriasis is usually in adolescence or young adult and tends to persist throughout life. The disease pathogenesis incorporates both mixed autoimmune and innate immune responses. And in about 20 to 25% of patients go on to develop psoriatic arthritis, which can be quite debilitating. And then there are multiple other comorbidities associated with psoriasis, including cardiovascular disease, metabolic syndrome, including type 2 diabetes. What about atopic dermatitis? It's one of the most common inflammatory disease of the skin. There's about 15 to 20% lifetime prevalence. It's also a complex genetic disease, multiple genetic variants, each one with a fairly small risk, but combined, they really help to kind of drive the disease pathogenesis, and we are up to over 100 genetic loci known to predispose to atopic dermatitis. And if you look at what those genes or the genetic variants are doing, they're really contributing and incorporating defects in the epidermal barrier, activation of different T cell subsets, and then there's dysbiosis of the commensal skin microbiome associated with this disease. It's a very heterogeneous disorder, variable clinical manifestations, and includes both acute and chronic presentations. And in contrast to psoriasis, the comorbidities that are associated with atopic dermatitis tend to differ quite a bit, and that includes food allergies, asthma, and allergic rhinitis being the most common ones. So if you think about, the histology of these two diseases, these are both papular squamous disorders, and that means that they're both involving the epidermis and abnormal epidermal function. And you can see that the changes on the plaque psoriasis end tend to be a little bit more and better defined than you can see in atopic dermatitis. But the immune networks that I have there on the bottom tend to be a little bit different, with psoriasis being kind of centered on IL-17, IL-23, and TNF-associated inflammatory mediators, and atopic dermatitis being different with IL-13, IL-22, and IL-31 being kind of the key ones. My background is in genetics, so I often kind of revert to genetics as a way to kind of look at diseases, and this is a kind of a diagram that kind of links together the different genetic variants identified with psoriasis with other chronic inflammatory diseases. You have psoriasis there in the middle, and then the different risk genes are kind of shown with smaller letters. But what I wanted to highlight is both the overlap of psoriasis with multiple other immune and autoimmune diseases, but it also has a bit of an overlap with atopic dermatitis, particularly involving the IL-13, IL-4 locus. And Dr. Brown is going to be telling you more about this in her talk later this afternoon. But I find it to kind of be quite interesting. If you look at this is what they call like a Manhattan plot of psoriasis. And you have the psoriasis meta-GWAS over here, and then you have the atopic dermatitis meta-GWAS over here on this end. And this is an old study. It was published about 9 years ago. But you can really see the peaks that you have in psoriasis sometimes kind of modeled a little bit on the atopic dermatitis end. Maybe not as strong. Here's the epidermal differentiation complex, much stronger in AD compared to psoriasis. And here you have the IL-13 genetic locus. But if you combine them, you get a very significant peak right here involving the chromosome location 5q31.1. This is the IL-13 gene locus. And it's actually interesting to think about it that in atopic dermatitis, you have an odds ratio with this locus of close to 1.3. But in psoriasis, you have the opposite. It's actually protective, about 0.86. So this is the conclusion that these authors made, that the results show that atopic dermatitis and psoriasis have distinct genetic mechanisms with opposing effects and shared pathways influencing epidermal differentiation and immune responses. So the variants predisposing to psoriasis tend to be protective for atopic dermatitis and vice versa. So it's this kind of like a mutual antagonism based, between these two diseases. This is a study that my group published about 7 years ago in the Journal of Investigative Dermatology, and it's based on bulk RNA-seq data. So what it shows you is that we're just comparing the two diseases on the transcriptomic level. And if you look at psoriasis versus healthy and then atopic dermatitis versus healthy, whether it's the upregulated or the downregulated genes, you can appreciate that there's much greater number of differentially expressed genes in psoriasis compared to atopic dermatitis. Perhaps not surprising if you look at the degree of epidermal changes that happen in these two diseases. And then you can kind of compare the overlap between psoriasis and normal skin, normal versus atopic skin, or psoriasis lesional versus atopic lesional, and you can appreciate the overlap, and then how the genes in psoriasis correlate with the ones that are differentially expressed in atopic dermatitis. And you can appreciate that it's actually quite similar. There's almost like a linear kind of a graph going over here. It's a little bit more skewed towards the psoriasis side than the atopic dermatitis side, but overall pretty similar. And if you look at the differentially expressed genes, about 81% of the genes that are differentially expressed in atopic dermatitis overlap with those of psoriasis. If you look at it from the other end, only about 41% of the genes that are differentially expressed in psoriasis overlap with those of atopic dermatitis. So these diseases have a lot in common if you look at it from the transcriptomic angle. What about the cytokine responses? They're definitely antagonistic immune profiles in psoriasis. This is a study of about 147 individuals. And you have the psoriasis lesional here in green, and then you have the atopic dermatitis here in the purple, and both lesional and non-lesional skin, and then healthy controls right here in the other corner. But what you can see is that the IL-17 response is what really defines and separates psoriasis from atopic dermatitis, where IL-13 responses dominate. And this kind of mutual antagonism actually is well described. This is a paper that was published in New England Journal of Medicine about 13 years ago. from the IREC group, and it's a patient that actually has both psoriasis and atopic dermatitis. This is the histology between the two. Here you have the psoriasis lesions, and then more like atopic eczema, atopic dermatitis. And then you can appreciate the clinical course where you're looking at atopic dermatitis and then psoriasis here in blue. And you can see that when one disease is flaring, the other one actually is getting better. And this is kind of like part of the spectrum that we often see with these patients, extremely rare, but we see them from time to time. And then you can see the histology is also quite different. IL-17 is much higher in psoriasis, and there's a little bit of it in atopic dermatitis. And you can see the same thing with IL-4 and IL-22, with IL-4 being more in atopic eczema, and interferon gamma being more higher in psoriasis, and IL-22 kind of being high in both, maybe a little bit higher in psoriasis. But it's also kind of important, and I want to highlight that, and we will have a talk on this also, is that the immune network in both atopic dermatitis and psoriasis actually is quite plastic. This is not a static kind of a diseases. There is quite a bit of shift, and we can see this when we treat patients with these very targeted kind of biological therapies. And it's well known that if you use either anti-TNF, anti-IL-17, or even anti-IL-23 agent, that a proportion of patients may actually go on to develop eczema-like lesions. And we're seeing more and more of this also on the other end. We have patients with atopic eczema or atopic dermatitis being treated with dupilumab, or even anti-IL-13 treatment, is that you begin, at least in some patients, to see shift towards more like a psoriasis-like phenotype. So it is quite dynamic, and this is still not well understood by the mechanisms involved. We have looked at this in our lab. This is based on the single cell data. You can look at the different immunological network, and the tools that we have today to kind of study this have really exponentially increased our chances to kind of take a really deep dive into the pathology. So if you look at CCL17 and CCL22, here we're comparing atopic dermatitis in red and psoriasis in green. You can see that these two cytokines or chemokines are higher in atopic dermatitis. But the ones that drive Th17 responses, such as CCL20, which is a chemokine for Th17 cells, or CCR6, which is the receptor on those cells for CCL20, that's much more dominant in psoriasis. You can also do the same thing for Th2 signal. You can look at IL-13, very strong in atopic dermatitis, barely detectable in psoriasis. But interestingly, the receptor for IL-13 and IL-4 are much more highly expressed in psoriasis skin. And what does that mean? We just don't know. And then also, if you look at the cell-cell interactions, this is just limiting it to the dendritic cell T-cell interactions. You can see the Th2 signal or ILC2 signal here is much stronger in atopic dermatitis and barely active in psoriasis, where the shift actually is towards other immune cell subsets. You can also look at this. This is just focusing on the dendritic cells. We have 12 subclusters of dendritic cells. You can separate that into 5 major clusters of dendritic cells, and then you can overlay the disease states to kind of see how they're kind of defined. You can see there's a little bit more psoriasis on this end and a little bit more atopic dermatitis on this end. These are the marker genes that you use to define these subsets, and then you can look at the cell numbers of the individual dendritic cells and see where it's actually skewed. And you can see that for the conventional dendritic cell 2A, they're much higher in atopic lesional skin compared to psoriasis. It's certainly a lot higher than you can see in non-lesional or control skin. You can look at them, and then you can use these data to kind of dig in even deeper. You can look at the differentially expressed genes in a particular cell type, which ones are increased or skewed towards psoriasis, and which ones are skewed towards atopic dermatitis. You can look at their expression of inflammatory mediators, as shown here in psoriatic skin, and particularly with activation of NF-kappa B and IL-8 or CXCL8. And then in atopic dermatitis, you're seeing much more of the CCL17, CCL22, and CD1B expression. And then you can use the tools that we now have to really kind of see how these dendritic cells are actually behaving differently and derived differently. So you have, these are all the dendritic cells in both psoriasis and atopic dermatitis. Here we have a conventional dendritic cell 2A, and here 2B, and then you have an intermediate state. And we can see that it's actually quite a bit of a trajectory where you have the CD2As on this end and the DC2Bs on this end, and then you can kind of compare how it the different chemokines are actually separating on this. You have atopic dermatitis here, psoriasis, lesion control. So a lot more dendritic cells in atopic dermatitis. The chemokines are really expressed here on the atopic dermatitis, the DC2A end, whereas the IL-8 and IL-1B is much more on the psoriasis end. And then you can see how these dendritic cells are really diverging in terms of their immune activation in these two different disease states, where it's going up in atopic dermatitis and down in psoriasis. And then you can look at differentially expressed interactions, looking at the ones that are enriched in psoriasis, which is the ones that are down in atopic dermatitis. And you can really see that it's the DC, dendritic cell interactions, that are really shifting towards being more active in psoriasis and shifting the balance with atopic dermatitis having more of a DC2A function and psoriasis more heavy on the DC2B end. And that's kind of shown over here. You can look at the different interactions. I'm not going to go into the details, but you can really see that the interaction between the two is really shifted between psoriasis and atopic dermatitis. And we think that this is one of the reasons why you're seeing this different polarization patterns in these two diseases, and also where you can have the switch between the two in these paradoxical type reactions. So just to briefly summarize, psoriasis and atopic dermatitis are two of the most common chronic inflammatory skin diseases. Both have overlapping histologic features and transcriptome, but they're characterized by opposite polar cytokine patterns. And while they're typically mutually exclusive, both immunologically and genetically, there are rare patients that have features of both atopic dermatitis and psoriasis. And the response in these diseases actually is quite plastic. And we can see these shifts with targeted therapies, but the mechanisms of those shifts are largely unknown. And then the regulatory mechanisms involved in regulating the polar T cell responses are largely unexplored, but dendritic cells, based on some of the data that we have, may play a key role in that part. Last but not least, I want to thank the IPC and the IEC for being here today and showing you some of the data. And then last but not least, I also want to thank the different funding sources that I've had over the years that have supported my work, and then my lab members and colleagues that I've worked with and collaborated on this. So thank you all for listening, and I think we may have some time for questions.
That's great. Thank you very much, Johan. We've got time for questions. Could I please ask that those who wish to ask a question identify themselves and their institution and country would be helpful. I can't believe there's no questions, so I will kick off. And it's fascinating, this single cell analysis. It looked a little bit heterogeneous in some senses, and I wondered whether you've been able to think about the different kind of subsets of psoriasis and AD that we think about, whether filaggrin-driven disease looks different to not, and so whether that alters the dendritic profile in that context.
Yeah, no, I agree. That's the problem with them, is that the level of heterogeneity, and that certainly is a lot bigger in atopic dermatitis than psoriasis. We see psoriasis being kind of less diverse. That being said, if you go into the clinical subtypes of psoriasis, you begin to kind of see these drifts, and some of them actually extend on a spectrum between an adaptive and more on the autoimmune innate type inflammatory responses. I think nobody has really compared, at least none that I know of, compared the filaggrin positive versus negative individuals in terms of like single cell type analyses. And I think that needs to be done. And also what we really need for practically both diseases is to try to link the genetics with the biology. There's still a major gap between our understanding of the genetic predisposition of those diseases and how those genetic variants actually play a role in the disease mechanisms. We don't know what cell type actually is primarily involved. We don't know if the genetic variant is activating those cells or making them more active or less active, and how the interplay between that. But we now actually have the tools to really dig into that. And I think that's where the field needs to go. But those types of studies are very expensive to run. And you need 200 or 300 patients to do it well. So I think it's coming, but we're a little bit off. And I think that's going to address the heterogeneity, because it's not just molecular heterogeneity, it's also genetic heterogeneity.
Yeah, and phenotypic. So I think one of the issues with this data, which we should encourage the community, is to be able to properly give the clinical phenotypes associated with the molecular data. because that's been limiting. I think everyone puts their data in relevant repositories, but there isn't always access to clinical phenotype.
Yeah, I agree. Access is critical.
Okay. Any questions then from the floor? No? If not, we'll thank Johan again. Thanks a lot. It's my pleasure now to introduce Michel Gillet, who I got to know well when I was on the ESDR board with Michel, who was subsequently ESDR president and is now program director of the EADV, and he's professor of dermatology in Lausanne. His research, as you know, is focused primarily on translational immunology related to inflammatory skin disease and melanoma, and he was involved in the discovery of TSLP, which is an important molecule, and particularly in atopic eczema. So without any further ado, remember you can always check his further bio on the program. We'll give us more time for talking.
Thank you, Nick. It's a pleasure to be here. So I will be talking about immune phenotype switching. I slightly changed the title of the topic. So I would like to start with a very simplified scheme here to show you again what are the key immune pathways which are important for us dermatologists. So I divided them in 5. As I said, it's oversimplification. But there is a number of pathways which are linked to adaptive immunity. So linked to T-cell differentiation, we have T-cells that can become Th1 cells producing interferon gamma, naive T-cells that can become Th2 cells producing IL-4, 5, 13, 31, as we heard. And then there are the Th17 cells, which produce IL-17 and 22. On the other hand, we have pathways linked to innate immunity, such as dendritic cells producing type 1 interferons and dendritic cells or other cells producing IL-1 family member cytokines, such as IL-1 beta and IL-36. So why are these pathways so important? Because they are directly linked to the pathogenesis of skin diseases, which we know. We know psoriasis, Th17 disease, atopic eczema, Th2 disease. We have other diseases which are linked to Th1, such as lichen planus. And then we have 2 diseases linked more to these innate pathways, such as lupus erythematosus, which is linked to activation of plasmacytoid dendritic cells to produce type 1 interferons, and neutrophilic disease, which are linked to the overexpression of IL-1 cytokines. We have very good targeted therapies which can block these pathways. They are very efficient, but we have always non-responders, raising questions whether we are targeting the right pathway, whether we have the right diagnosis, whether even we have some immune switches. And we performed a study to try to define these pathways molecularly in order to be able to adapt them onto the individual level and so search on each patient whether these pathways are actually activated. And to do that, we cross-compared the diseases that I've just shown you. We performed transcriptomics, differential expression analysis of one disease versus all the others in order really to be able to identify this key core immune pathway which is underlying the pathogenesis of this disease. And we performed nanostring analysis, and to make a long story short, here we identified the genes. We plotted them on this heat map on the y-axis here, and you see, to make a long story short, as I said, is that all the diseases, for example here psoriasis samples, they all cluster on an unsupervised way together based on the common expression of these genes which we identify here overexpressed in red on this heat map. They are all linked to Th17, so we call this the Th17 model. Atopic dermatitis samples, they're all clustered together based on the Th2 model, lupus based on type 1 interferon model, lichen planus based on Th1. Here we have some Wells disease, so eosinophilic cellulitis, which have an eosinophilic signature, and then neutrophilic disease, which have this myeloid signature, which we could divide into neutrophilic and macrophagic signature. So this is the basis of what I want to present today. So we have now a molecular cartography which allows us, on one hand, to diagnose disease. This is highly efficient. We can these sentinel diseases which I just shown you, we can diagnose them, but we can also diagnose less clear diseases, and I just would like to show you just a couple of examples. So we have an erythroderma case that was hospitalized 3 times. We thought it was a drug eruption, but when we performed nanostring clustered with AD, so it was an AD erythroderma. It had a dominant Th2 profile, as you can see here. So we took it, we clustered it in this cartography, clusters with the Th2, dominant Th2 profile, and it responded to IL-4 receptor, anti-IL-4 receptor therapy, so dupilumab. Another case, unclear case, very eczemative form in rash in a psoriasis patient, is the psoriasis AD. Again, we performed nanostring. It clusters in the middle of the psoriasis with the dominant Th17 profile, and it responded to IL-17. On the other hand, this one, is it psoriasis? Is it nummular eczema? I'm sure you in your clinic, you have these patients where it's not always very, very easy. So we performed nanoscreen. This clearly clusters in the middle of atopic dermatitis samples, has a dominant Th2. We gave it dupilumab, and it responded to therapy. So these cartography is highly efficient of helping us to diagnose these unclear cases. And also another thing we can do is to try to match the dominant model in this cartography with our treatment. So here I show you, for example, this is in atopic dermatitis. You see the dominant expression of the Th2 model compared to all the other models. So this would be matched to an anti-IL-4 receptor, so dupilumab therapy, but not to an anti-IL-23 or an anti-IL-17A therapy. On the other hand, this one, this is clearly a Th17 dominated disease. It's a psoriasis. This will be matched if you use an anti-IL-23 and 17 therapy, but mismatched if you use dupilumab. This is just to show you what I mean for matched and mismatched. And we did a study now with 80 and plus. We are now 100 plus patients. All the responders displayed matched models, so we had no clinical responder which had a mismatched model. However, if you look at the non-responding patients, we have 40% in the pretreatment biopsies which had a mismatched model. What was very interesting is that if we rebiopsied the non-responders, we found over 84% of the biopsies showed a mismatched profile, which is somewhat discrepant to what I've just shown you before. So here, this suggests us that there might be a switch of immune phenotype during the treatment, and this is the topic of my talk, and I would like to show you a few of these immune switches. So here I start with a 77-year-old male patient which has an adult onset atopic dermatitis, and he was given dupilumab therapy, but he flared after 3 injections, very eczematous, also on the scalp, on the back. So what is going on here? So in this patient, we had a pre-treatment biopsy, and we rebiopsied this flare. What did we observe? So the pre-treatment biopsy, it clusters in the middle of the Th2. It was an atopic dermatitis. It was the right diagnosis to put him on dupilumab. However, what did we see afterwards? So after the dupi, after the 3 injections of dupi, the flare of the eczema, we saw the decrease of the Th2 profile, but the emergence of a Th1 profile. And this is shown here also in quantification. So we see pre-treatment. The highest, the dominant pathway was Th2, and it switched to a dominant Th1 and a decrease of the Th2. We had several patients now with that. Here I show you 5 patients which flared up during Dupixent therapy. They all had a dominant Th2 here in blue before therapy. This is the pre-treatment biopsy. However, what happens after the post-treatment biopsy, when we had the flare, we had the emergence of this Th1 dominant pathway. So this is a switch from a Th2 to a Th1 dominant pathway. We know that IL-4/13 controls the IL-12 production. If you block it, we can imagine we have more IL-12, and this drives a Th1 response. What do we do? Therapeutic rematching, Th1 dominated, so we use the JAK inhibitor, which we know, JAK1/2 inhibitor, which we know can target interferon gamma signaling, and we had, after 4 weeks of baricitinib, a complete clearance of the rash. So another switch is this, what Johanna has already talked about, is this paradoxical reaction here. What we have is the dupilumab, so anti-IL-4 receptor-induced paradoxic reaction. So we have these psoriasiform skin lesions. In the literature, they occur between 2 to 6% of patients, so I'm sure you all also have seen that, so it's not so infrequent. Despite initial response to the therapy, here you see one of the clinical, one of my patients with the appearance of these quite well demarked plaques, scaly plaques. And so we performed, again, nanostring profiling. We see that untreated before therapy, this was a clear atopic dermatitis falling into the Th2 pathway. What happened in this lesion here, we have a clear Th17 pathway appearing here after 2 Apologize, it's the wrong way. So here again, the numbers, clear dominant Th2 pathway here, and after 4 months, starting Dupi, he developed these psoriasiform lesions, which were Th17 dominated. On the other hand, we have the reverse phenomenon. We have also psoriasis patients which are treated with anti-IL-17 therapy, and they can develop eczematous paradoxic lesions, so what we call paradoxic eczema. They occur in the literature is 5.8%, so also not unfrequent. They occur with all types of IL-7, Th17 blockade, so IL-17 blockade, but also Th17 blockade. They have been reported with anti-IL-23. Here, one patient, so untreated, clear psoriasis, Th17 dominated here. After secukinumab, switched to Th2 profile. Here are the numbers. Sorry, this is not the correct. So it was a Th17 to a Th2 profile. So what happens here? So we think we have a balance between the Th2, Th17 profiles. We know that Th17 through IL-17 controls somehow TSLP induction. If we block IL-17s, we have increased TSLP, which may drive then the Th2 profile. On the other hand, if we block IL-4, IL-13 signaling, we may have, again, more IL-23, which drives the Th17 thing. So we have a yin-yang between the Th2 and the Th17 pathway. Another paradoxical reaction, which is known and described since many years, is the anti-TNF-induced paradoxical psoriasis. This occurs in 5% of the patients treated with anti-TNFs. It's a class effect. Often it manifests. It can be pustular, palmoplantar. It can be on the entire integument. It can be often on the scalp. So we did profiling. These paradoxical reactions, they fall actually into psoriasis. They have Th17. But when we looked a little more closer and we looked at type 1 interferon expression themselves, we have a highly dominant type 1 interferon expression. We have described that years ago in 2015 with Kurt and Conrad. This is really dominated by type 1 interferon. How does this occur? We have described that. We know that interferon is crucial in the initial steps of psoriasis pathogenesis, can drive activation of dendritic cells and Th17 cells. These dendritic cells also produce TNF, which block then the ability of plasmacytoid dendritic cells to produce type 1 interferons. And on the other hand, they further enhance maturation of conventional dendritic cells to produce IL-23. So if we block TNF-alpha by an anti-TNF, this negative feedback loop is blocked, and these PDCs, these plasmacytoid dendritic cells, they produce constant, not inhibited type 1 interference, which drives a paradoxical reaction. So here, I show you again these numbers. So here, also Th17 dominate this anti-TNF paradoxic psoriasis, but type 1 interference. What is very interesting, and we are working hard on that at the moment, what we found, and this is a collaboration with Lone Skov from Denmark, we found that resistant psoriasis, they have a similar profile with very, very high dominant type 1 interference. This type 1 interferon production is even higher than Th17, so we have now the concept that this type 1 interferon is covering this Th17 pathway and is driving resistance to IL-23 and IL-17. I hope in the next future I will be able to tell you more. We have some evidence for the mechanism, but some work has still to be done. So, in conclusion, I think we have a immune balance. I've shown you these three pathways. So we have the Th17 pathway, Th2 pathway, the Th1 pathway, which are all interrelated, and they can be displaced by our biologics. So anti-IL-4 receptor dupilumab can go into Th1 or Th17. The anti-IL-17 can go into Th2, and the anti-TNF can promote an interferon, type 1 interferon-dominated inflammation. What is the consequence of that? We have resistant eczema, Th1 dominated. We have this paradoxic psoriasis, paradoxic eczema, which have somehow a flip-flop phenomena. They can go in one direction or back. And then we have the paradoxic psoriasis, which induced with anti-TNF, which is type 1 interferon-dominated. And finally, I think this will become very important, is resistant psoriasis, which are also type 1 interferon-dominated, and where the mechanism we still have to find, to describe it. I mentioned the frequency of those events, so around 5%, 2 to 6%, 5.8%. What is the underlying causing? We don't know. Is it genetics? I think it's genetics. We may have some epigenetics, some microbiome, but it will need to be determined. So how would we treat these immune shifts or paradoxic shifts? I showed you already for this resistant eczema, we chose JAK1 inhibitor because it targets interferon gamma, but I think we also should at the same time target the underlying disease. I mean, the risk if you target interferon gamma is that it switches back to the only. So far, we don't have any way to target interferon gamma signaling only, but we need to target multiple cytokine pathways. So the Th2 pathway plus the Th1 pathway, the JAK1 inhibitor. I think this is very important also in the paradoxic psoriasis eczema. Sometimes these patients do not require to go off their underlying biologics, and we just can treat it topically. But in more severe cases, I think it would be important to target both pathways, so Th17 through a JAK1/2 inhibitor, which targets IL-23 signaling and IL-4/13 signaling. Otherwise, you risk the flip-flop phenomenon. So if in a paradoxic eczema you give them dupilumab, the risk is that the underlying psoriasis will come back up. Same thing here for paradoxic psoriasis. For sure, in the context of a psoriasis, it would be optimal to target both the interferon signaling pathway, but also the Th17 pathway. And so these JAK inhibitor, TYK2 inhibitors would be the future for targeting these immune shifts. But a last example I wanted to show you is that we have also JAK inhibitor-induced paradoxic psoriasis, and we described that a couple of years ago with this patient who was treated with baricitinib for rheumatoid arthritis, developed these psoriasiform lesions on the scalp, and also these somehow pustular lesions, hyperkeratotic pustular, not very clear here. So we performed also nanoscreening analysis, and what we found is that it has a dominant Th17 pathway, but it has also very strong neutrophilic macrophages or myeloid signature. And why is that? We think that the JAK inhibitors, they're very efficient to target and inhibit multiple pathways. All these I've shown you, Th1, Th2, Th17, type 1 interferons, but they have no ability to interfere with neutrophilic pathways. And so here is again the scores for the pathways. We have a Th17 dominated, but also very strong neutrophilic dominated and macrophagic pathways. I'm showing here GPP. It looks very much like what we see in GPP. GPP also has Th17, but strong neutrophilic and macrophagic pathways, as you can imagine. The difference is in this paradoxic reaction, we don't have any type 1 interferons which are present in GPP. So with that, I come to the conclusion slide. I think I've shown you that there is a balance between immune pathways. Immune switches can represent the cause of non-responses. Genetics, that's my opinion, are likely to determine the predisposition to switch, but this needs to be proven. Molecular profiling may guide diagnosis and the treatment choice. JAK inhibitors targeting multiple cytokine pathways may represent a good therapeutic options for these switches. And with that, I come to the conclusion. Thank the people involved here, from my lab to the collaborators and my department in Lausanne, Switzerland. Thank you.
Thanks a lot, Michel. Any questions, please come to the microphone.
1346.
Hervé Bachelet from Paris. Very nice talk, Michel, as always. Can you comment more on your last slide regarding the sort of splitting between the neutrophilic dependent inflammation, which is not touched by the JAK1, JAK2 inhibition, because in transcriptomic analysis of GPP, but also palmoplantar pustulosis, you have some patients in microarray studies or others, molecular profiling, with strong MX1 or other interferon-stimulated gene upregulation, and others they don't. And you have the CARD14 dependent, the gain-of-function inflammation, sometimes leading to pustular psoriasis. So my question for you is, are there different ways to go to plaque, but also neutrophilic-dependent pustular-like inflammation, more than this very kind of, maybe simplifying separation between these two families? It's not a critic, of course. It's just to add something to the debate.
Yeah, of course, this was an oversimplified, and I definitely think there may be other drivers of the molecular phenotype which we observe in GPP and others. What, however, is very unique, I think, in all these paradoxical, what we call psoriasiform lesions, is they have a common Th17 seems always to be there to some extent. I mean, it's it's somehow decreased, but the Th17 always comes up like as a basis. I was surprised to find that, but as really a basis of this of probably a unique disease, which is psoriasis. And then the pathways which drive, or the mechanisms which drive these other additional pathways, they might be very distinct. I don't know if this is, I mean, you're also the expert, if that is what
no,
what you think.
Absolutely.
You think GPP is completely different? We always see this basic Th17 pathway, which might link it to psoriasis.
Thank you.
Thank you. Chris Griffiths, final question, please.
Hi, Chris.
Thanks, Michel. You and I have talked about this quite a bit before, and I guess you're a great advocate of next-generation healthcare, which is prediction rather than being reactive. how can we predict which patients are going to get these paradoxical reactions in the first place?
That's exactly what I wish I could tell you. We don't know, and that's why I'm saying I think it's genetics, but it's my hypothesis. I know that from transcriptomics I cannot predict. So we have these pre-lesions, and we try to predict whether they would switch Th1 or they would. We cannot predict. So we need to be more granular than these transcriptomic analysis. And I think we need to integrate genetics, we integrate microbiome maybe, we need to integrate a lot of these other omics technologies in order to go down to the essence, see what it is.
Thanks.
Thanks. Okay, thank you very much. We're just about exactly on time, so that's good. A good point to introduce Sarah Brown. Sarah trained in Newcastle and subsequently has moved to Dundee and then has recently become the Grant Chair of Dermatology at the University of Edinburgh and is also a prestigious Wellcome Trust Senior Research Fellow. She's had a long-standing interest in atopic eczema and her lab is really focused on functional genomics. So we look forward to your talk, Sarah.
Thanks, Nick, and thanks for the invitation to speak, especially after two such informative talks. It's made my talk easier. So this was the title I was given, Genetic and Ethnic Differences in Psoriasis and Atopic Dermatitis. This is what I'm hoping to cover in the next few minutes. So firstly, just to pause and talk about the nomenclature of race, ethnicity, and ancestry. It's a technical detail, but I think it's important. Why does genetic makeup matter? We've just heard genetics is likely to give us a clue to causality. And why is it important to understand these differences? And then a little bit about psoriasis, atopic dermatitis, and then focusing on what further work we need in this field. So I think it's important to think carefully and clearly about the difference between race and genetics. To avoid any kind of confusion or upset that borders on racism. So race and ethnicity can be defined in the Cambridge English Dictionary. As you read here, it's a group of people who have these things that link them together, a state of belonging in a group. And it's socially determined and self-identified to a certain extent. We do ask people what race or ethnicity do you identify with. And then if you compare that with ancestry, again, this is the Cambridge English Dictionary definition, not necessarily useful in the context today, but the important thing about ancestry, it's genetically determined. So you can't choose your ancestry, it's something that you inherit from your parents, not socially determined. But actually, in the context of skin disease, both of these matter. And I'm pausing to go over the nomenclature because I think it helps us think very clearly. Are we talking about race and ethnicity, or are we talking about ancestry? Because actually, they both correlate with each other, but they kind of tell us different things. So your race and ethnicity are tied up with how you behave, where you live, how you look after your skin. Your genetic ancestry is kind of the nitty-gritty, how your skin was made. And obviously, to understand our patients and to understand their skin disease, we shouldn't just focus on one or the other. We should try and understand both race, ethnicity, and genetic ancestry. So how do we apply this then to psoriasis and atopic dermatitis? So again, in the previous talks, we've been shown beautiful pictures of psoriasis and AD and how they're similar in some ways and different in others. And it's very interesting to see that the clinical presentation is different again in people with skin of colour. So some people say it's harder to discern the erythema in people with skin of colour, especially very dark-skinned people, because the erythema can be kind of violaceous, this sort of purpley black colour, instead of the red, the bright redness that we see in white skin races. Another important feature is that, in any skin we can get post-inflammatory hypo or hyperpigmentation, but these can be particularly obvious and sometimes clinically more challenging in people with skin of colour. They have more colour to lose, but they also seem able to have more colour to gain. And it probably is something to do with the inflammatory mediators that can inhibit melanogenesis, but they can also, the same inflammatory mediators can also stimulate melanocytes to divide. And then there are various clinical mimics that can confuse the dermatologists in relation to psoriasis, especially psoriasis in skin of colour. So I've just listed a few here. So then what about atopic dermatitis? This also looks quite different in skin of colour. So again, you can list some of the same challenges. The erythema may appear violaceous. You might get more prominent post-inflammatory hypo or hyperpigmentation. These are challenges shared with psoriasis. But in addition with AD, you get this very, very different papular or follicular pattern. I remember clearly as a junior dermatologist the first time I was shown a patient with skin type 6 who had atopic dermatitis. I couldn't believe it. It looked so different from the AD that we'd seen, that I'd been taught to look for ill-defined erythema and scale. It's kind of the opposite when it comes to that picture on the top left. This is a very papular or follicular pattern. You see it there on the abdominal skin top right. And then the other thing that you get with atopic dermatitis in skin of colour is more prominent lichenification, and we're not really sure why that is. So then looking at the epidemiology of psoriasis, this is a quote from the Global Atlas of Psoriasis, which is a phenomenal resource looking at psoriasis across the world. You can see that it is more prevalent in northern latitudes than the western countries. The colour coding here is the psoriasis prevalence in percentage. But even within the US, which is coloured all the same way, there are ethnic and racial differences. So Caucasian people have a prevalence of psoriasis that's substantially higher than the African Americans, which is higher again than the Hispanics. And these are people groups living within the same cities. And it's also interesting to know that ethnicity may affect severity. There's obviously multiple reasons that this could occur, either genetic or environmental. And this was a retrospective study looking at about 800 cases, of which the Asian people had more severe psoriasis than the white-skinned people. The Hispanics had more severe than the white. And unfortunately, African American and Middle Eastern groups weren't adequately powered to make any real conclusions. But obviously, the severity can affect the diagnosis of disease, so it might also impact on the apparent prevalence. So then what about the epidemiology of atopic dermatitis across the globe? In many ways, it's similar to psoriasis. You see it's tending to be more common in northern latitudes, tending to be more common in some of the western world. And this is a publication that was produced by the International Eczema Council, led by Katrina Abuabara, who linked the prevalence with also climate hazards. And we did a systematic review, but then looking at data that was available of AD cases compared to this environmental hazard index. And you can see that the cumulative exposure to climatic hazards, multiple different hazards that might increase the risk of inflammatory skin disease, you see there is a very rough overlap. So coming back to genetics, can we use genetics to understand the difference in psoriasis in people of colour? Well, the major challenge here is that by far the vast majority of studies have been on white Europeans. And that's partly because psoriasis is much more common in white Europeans, still relatively rare in other populations. But it is common enough in Han Chinese that we can compare between the Han Chinese and the Caucasian. And this was a study published in 2015, and I've deliberately made the graphs a bit too small for you to read, but hopefully that you can see in the Chinese population there's one massive peak, in the Caucasian population there's one massive peak, and when you put them together there's an even more highly significant, that's a p-value to the minus 265. It's ridiculously statistically significant. So it's fair to say the HLA locus is the main genetic risk, and within that it's this allele HLA-CW06. And it's simple, but this same paper noted importantly that there were diversities between the populations. So it's HLA-C that is shared. HLA-A, this allele here, is prevalent as a risk factor in Chinese, but it's very rare in the European population. So it's not just that it's not so much that it doesn't cause psoriasis in the Europeans, it's that it's not there in the Europeans. Conversely, the HLA-B locus comes up in the Caucasian people, but it's very rare in the Chinese populations. But the other genetic associations were also heterogeneous. So these other peaks that are a bit lower down, they're harder to see, but they also showed population-specific effects and what we call allelic heterogeneity, which means there's a locus that comes up in the same populations, but when you map it in detail, the different variants within that loci acting in the different populations. So it genuinely is worth doing these studies across different populations. We learn a lot about the nitty-gritty of the genetics, what might actually be causal. So coming more up to date now, a group from the US did this larger transethnic analysis of psoriasis, this time with South Asians and Europeans. And here again, that same HLA-C allele was the top-ranking MHC locus. And fine mapping across the population suggested that the protein might actually have a causal role in disease. But again, the advantage of getting a more diversity within the genetic ancestry is that you can fine map other signals. So you're not just getting a single massive peak because of one effect, you're driving a massive peak that when you dissect it, there are actually multiple effects coming together to raise that signal. And you can start to separate these when you have enough genetic diversity to be able to do that. There was a very nice review from Nick Dand and colleagues in ACTA-DV a few years ago, and they just commented there'd only been quite small studies in these other ancestry groups. And sadly, there are no major studies that I could find anyway in psoriasis from people of African ancestry, apart from some African American people. And again, this probably reflects in part that psoriasis is relatively rare in Africa, but just to emphasize it is a real gap in our knowledge. So what about genetics of atopic dermatitis? And Johan already presented work that we did quite a few years ago, comparing and contrasting AD and psoriasis. So I'm just going to focus on the AD data just now. So this is our equivalent of the meta-analysis in AD. We had a very large discovery cohort and an even bigger replication cohort. Again, we called it multi-ancestry, but rather dominated by people of white European ancestry. But we did manage to get people of Japanese ancestry, African Americans, Latinos, and some people of mixed ancestry from an Australian cohort. So putting these together, again, you're familiar with Manhattan plots. The biggest peaks show the most highly significant regions of the genome that are associated with the disease. And just eyeballing the European compared to the multi-ancestry cohorts, you can see that they're pretty similar. So the themes here are skin barrier and inflammation. Won't come as a surprise given what we've heard already. The main skin barrier peak is that one on chromosome 1, which is the epidermal differentiation complex, including filaggrin and related genes. And then the second biggest peak is on chromosome 5, which includes the major histocompatibility complex. So putting these together, we found a total of 81 loci, of which 29 were novel because of the bigger data set in the European-only analysis. And we found 15 additional loci. Sorry, my text has jumped around a little bit. When you add in the multi-ancestry people. So again, it's emphasizing that, sometimes in genetics we try and make it a single phenotype or a single ancestry, but in this context it's particularly helpful for it to be a diverse ancestry to find more information. And looking at, again, these regions of DNA, they're just regions of DNA. Many of them are in between genes, so it's the start of a long pathway to try and understand what they are and what they do. But one step in that pathway is to look at whether, relating to the question that we had in the beginning about which tissues are important. So one way we begin to find out which tissues are speaking this genetic code, we look at where the DNase 1 hypersensitivity is. That means the coiling of the DNA is opened up, ready for those genes to be expressed. So where the loci are open, ready to affect gene expression. And you can see in a kind of a humbling way for a dermatologist, I like to focus on skin, but actually this analysis showed us that the DNase hypersensitivity are overrepresented in blood. So yeah, annoying when you're a dermatologist, but also very good news because it means we can study blood and find out important things about gene expression in these systemic inflammatory disorders. So coming back to molecular mechanisms, again, the previous talks have described these really beautifully. I couldn't find in my literature search preparing for this talk any studies comparing the different ancestries with all the beautiful molecular data that we've got. Again, I'd be delighted to hear from the audience if I'm wrong. In molecular mechanisms of atopic dermatitis, Emma Gutman-Yaski has really led on this concept of endophenotypic variations using transcriptome analysis to compare populations within the US. So these are white-skinned European people, people with African-American ancestry, and people with Asian ancestry. And that group has reported that the European-American patients, as we've already heard, have a predominantly Th2, Th22 activation with less of the Th1, Th17 axes, and this common suppression of filaggrin, whether they have loss of function mutations in the filaggrin gene or a down-regulation in gene expression because of the atopic inflammation. And then Asian patients are somewhat different in that they have even more accentuation of Th22 and a bit of a Th17 pathway. They also have epidermal barrier defects, and depending on which Asian population you look at, some have a complex pattern of many different filaggrin mutations, others seem to have filaggrin or other epidermal barrier genes suppressed by the inflammation as a secondary effect. And then in African American patients, there are really very few filaggrin mutations, and there seems to be an attenuation of Th17 and Th1. So really quite a different immune phenotype that could be contributing to the differences that we're seeing clinically. And this is just a shout out for a review that a junior colleague of mine in Singapore has written. So this is in press now at the JID, comparing and contrasting what is known about atopic dermatitis in the white European population with this really interesting population that they have in Singapore that represents Asia, in that it includes East Asians, Southeast Asians, and South Asians, all together within one population. So exposed to a certain extent to similar environments, but they have different prevalences and different types of atopic dermatitis. And in this review, he's a clinical academic, and he writes really nicely, I think, about these different aspects of pathomechanisms. So you can look out for that coming out soon. So many of us in the audience are clinicians, and we're very interested in treatment response. And again, the last question from Chris Griffiths, can we predict how patients are going to respond or not respond, or worse still, get paradoxical response? We're still struggling with that genetically. What we do know is that this HLA allele gives conflicting effects in response to anti-TNF therapies. Some studies have shown it improves your response, others have shown it reduces your chance of response. Conversely, there is more consistent evidence of association of a better response to ustekinumab if you carry this common risk allele. But there's lots more that we need to learn. What about atopic dermatitis? We don't know much more, but again, the data that is there shows that there might be a differential response to systemic immunomodulatory treatment in patients with dark and light skin. In this study of about 200 patients, just again a retrospective observational study, But here on the left, this is people with dark skin tone. So this is not defined. This is not self-defined ancestry or ethnicity. This is not self-defined ethnicity or genetically defined ancestry. This is kind of Fitzpatrick skin type, really. So dark skin is in this red part of the graph. Lighter skin people are in the blue part. So what you're seeing is in these people, when they're treated with dupilumab, the people with darker skin tones respond quicker and seem to get a better response. We didn't see any differences with methotrexate and cyclosporin. The grey is the overlapping confidence intervals. So it's not a dead cert. There's a lot of overlap, and it's retrospective data, etc. There are biases in the patients that were chosen to receive dupilumab, obviously, because it's observational, so-called real-world data, but it's definitely something to consider. And we've debated quite a long time in centres that use a lot of phototherapy, whether people with darker skin tones might respond more or less to phototherapy, because with our algorithms, if you've got a darker skin tone, you're allowed to get more phototherapy, but you might be more resistant to it. It's not really clear. So in conclusion, I hope I've convinced you that it's important to understand these similarities and differences between the genetic ancestries and the social ethnicities that we have. It's important because people have different skin phenotypes in response to the different diseases. Genetic mechanisms probably contribute to those phenotypes, but we need to understand them better. Environmental effects that are very different in different societies clearly contribute to the phenotypes, but we need to understand them better. And phenotypes almost definitely affect treatment response, and that's really a driver for understanding all of this. Looking further afield, the burden of disease is different in different ethnicities, and we need to keep that in mind, partly related to the phenotypes and how different people respond to those. And then barriers to healthcare is something that I haven't even touched on, but it's a huge part of our work and interest that we need to keep in mind. So lots of ongoing work and lots of future work needed. It is very urgent to include greater diversity in all our research. We need more mechanistic studies in skin of colour, because ultimately we must provide personalised medicine for different ancestries. And we're by no means alone in trying to do this. I'll just leave you with this quote: "The importance of sincere, concerted global efforts towards genomic equity is something that I think we can all sign up to, not just in dermatology, but across all of genetics." And thank you very much for listening.
Thank you very much, Sarah. And it's very pertinent, your call to arms, given the mission statements of both the IEC and IPC. And I think it's something maybe the boards could consider, how we could come together as a community to try and support these larger scale studies. Any questions for Sarah? Okay, well, I'll ask one then. Well, in terms of, I mean, it was fascinating that you're in AD, that you found the EQTL were predominantly in blood. So, which does make it much easier then, doesn't it, to go forward and do large-scale samplings. We've all been talking earlier in the day about integrating either single or bulk RNA-seq with genetic data, and it has to be done at scale in hundreds of people, really, doesn't it? So that is challenging if you've got to get skin biopsies from everybody, but if you can do it on blood, it's much more doable. Mm-hmm, mm-hmm. So where do you think the community is with that?
Yeah, I think, like you say, in some ways getting blood samples is easy. In other ways, it's not easy, is it? Because it still takes massive coordination and massive working together. And I think the easier, the more we do that, the bigger numbers we're going to get, and the more we're going to be able to address the difficult questions. I think at the end of the day, blood is never going to give us the whole story. We know that the skin is intrinsically important. So for me, it's about matching together what we find out from blood and those very big numbers that we get with a more detailed phenotypic understanding from the skin. And I guess that ties in with a comment you made about sharing clinical data. So we know details like what phase of the disease was this skin sample taken from, this blood sample taken from. So we can start asking those really, more detailed but more important questions. So yeah, blood is useful, but it's never going to give us more than literally a circulating readout. If we want to understand the real what's going on in our organ that is giving us the clinical readout, then that is what we will still need to sample.
Great, thank you.
So, Krasagakis from Crete. Thank you for your excellent lecture. I have a question and a kind of question. I think that the published studies on the genetic difference in atopic dermatitis are based on the fulfillment of the criteria, of the classic criteria of atopic dermatitis. But what happens when patients, for example, in Crete, we have very few patients that fulfill these criteria, but we have enough patients that develop eczematous skin reactions in older ages that the only thing that we found is an atopic background. For example, eosinophilia, IgE responses, atopy of other types than dermatitis, atopic dermatitis. So what do we do about these patients? And this is, I think, very important because all studies about new treatments consider the fulfillment of the classical criteria. So we cannot prescribe them as cytokine therapy, for example, anti-cytokine therapy.
Yeah, so it's a very interesting question. I mean, epidemiology is teaching us that there are a large proportion of cases that can be truly adult onset. And I guess that's what you're describing. It's atopic dermatitis, but it's truly adult onset. So, yeah, in terms of genetic studies, we are looking now at adult onset atopic dermatitis to try and understand that. To see if it's distinctly different. I suspect it won't be. I suspect it'll be, a mechanism that for some reason enough of the burden of inflammation and skin barrier breakdown just occurs later in life. So it's probably more of a regulation or an ethical question, whether those patients that, just because they weren't lucky enough to get atopic dermatitis as children, they're unlucky enough to get it as adults, they should still, in my thinking, if they've got essentially the same disease, be eligible for the same treatments. And I wonder if it's where, Michelle's molecular phenotyping will come from. So as a clinician, we might not be able to just fit them into the UK Working Party diagnostic criteria, but the proteomics or transcriptomic profile will fit them in there nicely. That surely is the future.
Okay, great. Thank you so much. So we've got now a panel discussion. So I'd like to invite the panel to come back, please. And also the panel discussion is going to be led by Tiago Torres, who I'd like to introduce, who from Portugal, who's Professor of Dermatology at the University of Porto, and also leads both the Psoriasis and Eczema services at the University Hospital. So he's perfectly placed to lead the further discussion. So we've got about 10 minutes for this. Could you please put the lights up and then we can have a further discussion and explore some of the themes that have started to come out. I hope we've got enough chairs.
We've got these slightly scary.
You can sit down.
Definitely not.
You sit down then. I'll sit.
You sit on one of those, mate.
I'll sit on one.
No.
You sit down. Yeah, it's fine.
There's enough.
What a relief. I was a bit scared about those chairs.
Okay, thank you. So we have some minutes for discussion now. I think we can continue with any question for any of the speakers from the audience, if there is. Okay, so I would make a question to Sarah about the management of non-white patients. At least the data from psoriasis trials in specific non-white population, like Visible with guselkumab or even Subanalis in other clinical trials, does not seem to exist a difference in terms of efficacy in white and non-white. Do you think that is because there's not enough differences in terms of pathogenesis between the two ethnicities? And what do you think in in atopic dermatitis? Because probably there are more differences, immune differences between these two populations.
Yeah, I agree. In AD, the immune system seems more diverse within people of colour. I must say, I'm not an expert in treating psoriasis. My specialist interest is in eczema genetics. But my interpretation of the lack of difference in response would always be, are we not seeing a difference because there genuinely is no difference, by which I mean people of colour respond to treatments exactly the same when they have psoriasis as people of white skin? Are we not seeing a difference between the ancestries because there is no difference, or because the studies have not been adequately powered in terms of including enough patients? So that would be the key question. From AD, where I do have more experience, yeah, I agree. I think, again, Michelle said that AD is a more diverse disease than psoriasis immunologically. I think it is, in terms of barrier structure, it's more diverse as well. So I'd have every expectation that people of different skin types would respond to different treatments. It's just going to take time for us to understand those and target appropriately. It's really a steep learning curve, and it's more emphasis on recording diversity. You know, when you look at studies, sometimes it's not even recorded the ethnicity or the ancestry of the patients in the very big population studies. So we do have a long way to go.
Okay, thank you. So any question from the audience? Oh, yes, there's one.
Yeah, I can maybe add to it.
There's one. Sorry. Oh, okay. Yeah.
Sorry, I don't know if you can hear me. Shobha from Newcastle. I had a question for the phenotype switching person, Michelle Gillette. Your resistant psoriasis cohort was very interesting. And I wondered if you actually saw any sort of Th22, IL-22 signature. That was one of the questions. The other question I have for the panel is, there's quite a lot of Th17 T-cell data there. How are these various phenotypes affecting your macrophage myeloid, the innate immune cell populations? I presume, it's very hard to do that with RNA-seq. But I just wonder, how is this, for example, in the resistant cohort, all these cytokines such as IL-17 or IL-22, how are they affecting the innate immune cell population? That would be my second question. The first question is, do you see a lot of Th22?
So there are very good questions, but we have not looked into detail whether IL-22 is upregulated. But from what I recall, I mean, there was no obvious signal, but I need to go back to look into details. So we have not done extensive studies yet concerning your macrophage myeloid effect of this resistant patient, but it's of course very interesting. I mean, I can. It is not true because we have one of the mechanism we think that these. Well, I've shown you in the resistant psoriasis type 1 interferons, which are, and we have a huge cohort together with Copenhagen, with Lonnie Skov, and we see type 1 interferons are really a key signature. And we think it's a lack of control by macrophages on type 1 interferon production, and we have a mechanism.
Dendritic cells.
Yeah. Yes, from plasmacytoid dendritic cells.
Dendritic cells, yeah.
So we think macrophages are very important myeloid cells to control type 1 interferon production by dendritic cells.
Right.
So we are looking into, whether it is an epigenetic modification of macrophage. I hope I will be able to tell you more soon.
Thank you.
But it's the way we are going, yeah.
Okay. And with the Th22, there's just no signature with protein at all?
Yeah, we have not looked into detail. I cannot say that this is, but maybe I should look at the data.
Thank you.
Thank you.
Okay, sorry, Johan, I just interrupted you. You were going to, I think, comment on the previous one. Sorry.
No, I was just going to add on to what Sarah said, that, the racial differences are obviously massively understudied, but there's some evidence from clinical studies, like, for example, African Americans with psoriasis tend to have more severe disease. And there definitely is a link to the genetics through that. We see increased or high frequency or carriage of specific risk genes involving NF-kappa B and IL-17 response signals. So I think, with appropriate studies, obviously they need to be of certain size. I think we're going to be able to address a lot of this. And then the other thing that we haven't heard yet is actually that I think is also important is the sex differences in these diseases. And it's not that they're pretty close to being equally common in men and women, and I think atopic dermatitis might be a little bit more skewed towards higher incidence in women.
Well, AD is really interesting in the early stages of like early childhood, it's more common in males and females, but then later on it's more females than males.
Yeah, and I think that's an aspect of the disease that is still very understudied. In psoriasis, we have equal frequency of the disease in men and women, but women are much more likely to fail biologic treatments, that they fail them more quickly than men. And men tend to have, so much more severe disease at baseline. So I think there are these tweaks in the pathogenesis and the mechanisms that we just don't understand and we haven't really addressed yet. So I think there's a lot to be done, and it goes beyond race and ethnicity also.
Okay, thank you. Nick?
Yeah. So my question follows nicely on, I think, from that, in that we also know that environmental factors, including body status, BMI, massively influences response, I think, for both diseases. And it's been underappreciated probably for AD, but it's certainly a factor. So I wondered, in this complex immunological milieu, how people have hypothesized for years, haven't they, about how BMI may influence it, but there isn't that much data as far as I'm aware. But useful to have a debate about environmental influences on clinical presentation, response, and molecular classification. Of silence.
It's a very big question, Nick. You've stunned us all into silence. I think from a genetic...
I think those Ozempic studies are coming, and I think certainly in psoriasis. And I think more likely than not, they're probably going to work.
Okay.
From a genetics perspective, it's super challenging because it's like, you saw the size of the GWAS, that's like 1 million different SNPs. And then if you add in environmental factors, you can add in however many more environmental factors you want to. So say you added in 10 environmental factors, then it's 10 times the number of tests that you have to do with 1 million SNPs. It's 10 times 1 million. So it just, in terms of the burden of multiple testing, this concept that the more investigations you do, you find positives by chance, and they could be false positives, it becomes statistically very, very challenging to really address all the different environmental factors with all the different genetic factors. So the approach I'm taking in my lab is to model it in vitro, which again is the other extreme. You take one gene and one environmental factor and try and dissect what's happening, and that's really painfully slow progress, but it's complementary to the gene-environment interaction analysis that we're doing in silico. Because it is really important, again, in clinic, patients say, if they've had one child with an inflammatory skin disease, it is a fear that they'll have another child with an inflammatory skin disease, and they ask you, what can I do to look after my next baby? And it's, should I get rid of the cat? Should I buy a dog? These are really important questions in the real world. Whether or not you get fat or whether you manage to lose weight is another very important question. And I think from Mendelian randomization, so again, a genetic approach to look for causality, there is evidence that increased body mass index has a causal effect on psoriasis and, to a lesser extent, atopic dermatitis. So we don't know what the mechanism is, but we do know it appears to be causally related, not just epidemiologically associated. So even more reason not to be overweight or obese.
Okay, thank you. Well, we're running out of time, and I don't want to shorten too much the break. So I think we can interrupt here. We'll have 50 minutes of break. Thank you. Yeah, thank you. Oh, no, it's okay. Okay, so welcome back to this joint symposium, IEC, IPC. And now we'll have 6 short presentations. And then we'll have some time for Q&A for each presentation. I would start calling Anna Rebane from University of Tartu, Estonia. And we'll address epigenetic and transcriptonic characterization of skin oming CLA plus CD4 T cells in atopic dermatitis and psoriasis.
So first I'd like to thank organizers for this opportunity to give a talk about our work. And as you can see, we work on CLA positive CD4 T cells in AD and psoriasis. And our work is part of IMI project called Biomarkers in Atopic Dermatitis and Psoriasis, a big consortium. So probably everybody knows that T cells with cutaneous lymphocyte-associated antigen, CLA positive T cells, are able to circulate between skin and blood, and we took advantage of it in our work, and we are working with blood purified CD4 CLA plus CD4 plus T cells. And this group of cells is actually heterogenic, but people sometimes forget it, that they also have lots of Tregs among them, but we have all bulk everything there. So our general workflow was to purify PBMCs out of patients of atopic dermatitis, psoriasis, and LT control, about 10 people in each group. Then we did FACS sorting for CLA positive T cells and for control naive CD4 T cells, and some of those T cells were subjected to epigenetic analysis using so-called ATAC-seq, which enables to look which areas of the chromatin are open. And another part, we did just RNA sequencing, and we received different numbers of cells that we didn't optimize. And when I go back, these 2 people are very important in this work. They have done most of the work. Regina did all cell purifications and sorting, and Martin has done all data analysis. What we see from our results is when we look principal component analysis is that when we look RNA-seq results, AD patients grouped differently than psoriasis patients, and controls are even more separate. When we look CLA positive T cells and even naive T cells, with ATAC-seq naive T cells and CLA positive memory T cells, they distinguish really well, but not in case of patients. They are actually quite similar. So the epigenetic, when we look at chromatin opening level, very few differences. And if we go more deeply in the data, we really see that in ATAC-seq there are over 14,000 different peaks. When we look just naive cells and CLA positive cells, but as soon as we start to look differences between disease and control, only atopic dermatitis has slightly more differences than psoriasis. In psoriasis, we were able to detect less. Interestingly, the same is when we look transcriptome, we have more differences between AD and control, and as psoriasis and control. We don't know exactly why it is like it is. What is not written in this picture is that actually there is overlap between AD between those genes which are differentially expressed between AD and control and psoriasis and control, the overlap is about 1,000 genes. So 1,000 genes are overlapping, which are different in both diseases. But when we started to look whether RNA-seq and ATAC-seq give overlap between each other, we found only very few overlapped genes. So it seems that the differences at the chromatin level are slightly more far from the differences at transcriptome level, which is more or less reasonable. So we also have looked using reactome which pathways are influenced on RNA-seq data, and I come back to the result just slightly later. And then we have also looked what kind of whether when we look at ATAC-seq data, we have some differences. And then we looked if there is a difference, whether this is associated in any transcriptor factor binding sites. And what we see is actually that there is a nuclear factor of activated T cells, which is controlled by calcium and calcineurin, and which is also, for instance, enhanced for instance, NFAT-C knockout mice, the DH1, DH17 pathway is enhanced. So this is transcription factor which clearly modulates T cell transcription profile. And psoriasis, we can see that there maybe its family is influenced. Again, taken everything together, if we look the transcriptome, we see differences transcriptome in this part. In IL-4 and IL-13 signaling, maybe IL-17 signaling, in AD CLA positive AD CLA cells, and in psoriasis there is interferon signaling seems to be affected in our case. And both CLA, both diseases are characterized with increased proliferation of CLA positive T cells, which is kind of reasonable because you have disease and you have more proliferation, more active T cells. So conclusions are that we see that CLA AD T cells are more affected than CLA psoriasis cells. We see maybe perhaps increased proliferation, and we see some particular transcription factors to be related. And here I thank all my group, and certainly Martin and Regina, and clinical collaborators. We got the blood samples, and all IMI consortium, and other funding bodies. Thank you.
Thank you. Any question? Okay, no question. How do you think this can have implications in the future management of the diseases? Do you think this...
There is no systemic management of those patients we selected. And even during the past recent weeks, there is no management yet.
Okay.
Yeah. So, but what exactly?
No, I was asking how do you think these results may have an implication in
how our results may have implication to the management. I think what we can see is that CLA positive T cells are really important because the transcriptome is changed and they should be targeted. It's maybe one of the ways how to target the disease is target specifically CD4 T cells, CLA plus CD4 T cells.
Okay, thank you. Thank you so much. Okay, so we would continue now, and I would call Alison Trischel from Cleveland, Ohio, and will address protective effects of systemic therapies for psoriasis against thrombotic events. Thank you.
Good afternoon, everyone. I'm excited to be here today, and I'd like to thank the organizing people for giving me the opportunity to present today. We have no relevant disclosures. Psoriasis is associated with an increased risk of cardiovascular disease, and it is hypothesized that the mechanisms underlying psoriasis and the associated chronic systemic inflammation accelerate the development of CVD, including thrombotic events. However, it is unclear if systemic therapies that treat psoriasis also reduce the risk for thrombotic events. So there are several challenges to studying CVD outcomes in psoriasis. First of all, you need a very large patient population to have the power to detect differences in CVD events. You also need a long follow-up period to allow for enough time for events to occur. And there's also several confounding factors like comorbidities associated with psoriasis, as well as medications like statins that could impact your outcomes, which can complicate your analysis. And of course, all of this could get very expensive. So our aim was to utilize the Trinetics database to investigate the effects of systemic therapies on the risk of thrombotic events in psoriasis patients compared to those treated with topical corticosteroids within 5 years of initiating treatment. So to investigate this question, using the Trinetics database platform, this includes over 100 million anonymous patient medical records in the United States. We're able to adjust for age, sex, weight, race, several comorbidities like CKD, alcohol use, smoking, as well as medications like aspirin, antilipemic agents, and anticoagulant use. We identified 700,000 patients with psoriasis in the database diagnosed with psoriasis between the ages of 18 and 80 years old. We excluded 500,000 of those because they were not on single agent therapy during our study period. So then we pulled out groups who were on systemic therapies as well as a group on topical corticosteroids. And this is people who initiated treatment with this agent within 1 year of their initial psoriasis diagnosis. We then propensity match scored each group of systemic therapies to topical corticosteroids. So we had one systemic therapy patient in each group that was matched to a topical corticosteroid patient. So this is a complex slide, but I'll guide you through with some arrows. First, let's look at the deep vein thrombosis. We saw that IL-17, which is in green, and the IL-12/23s or 23s in gold, had a protective effect relative to topical corticosteroids as they're to the left of the dotted line, whereas methotrexate in blue and is to the right of the line, meaning there is an increased risk of DVT. Next, looking at acute myocardial infarction. This was reduced for the IL-12/23 or 23 group as well as apremilast. And then looking at lab outcomes, I'll just highlight that methotrexate, again in blue, is to the right, meaning that there was an increased risk of thrombocytosis or elevated platelets relative to the topical steroid group. So to summarize the findings, methotrexate was associated with an increased risk of thrombotic events as well as thrombocytosis. Apremilast and anti-TNF-alpha had some protective effects. IL-17 showed a reduction in several thrombotic events. and the IL-12, 23, and 23s protected against the most thrombotic events as well as new onset thrombocytosis. So in conclusion, biologic treatments could potentially reduce the thrombotic events, and there may be differences in the protective effects between drugs, but we're not powered to detect differences between drugs for this study design. Methotrexate was associated with an increased risk of thrombotic events, and further studies investigating the systemic therapies of CVD and psoriasis and the underlying mechanisms is warranted. So I'd like to thank Dr. Kevin Cooper, along with the rest of our lab members, for their help on this project. And I'd also like to thank the IPC for the opportunity to participate in the International Fellowship Program. And after this meeting, I'll be heading to Madrid to work with my IPC mentor, Dr. Gonzalez-Cantero, who we actually had the pleasure of hosting a couple weeks ago in Cleveland. And we look forward to establishing our international collaboration with him. And I can take any questions at this time. Thank you.
Thank you. Thank you. We have a question.
Thank you very much, Nick Reynolds, Newcastle. Very interesting data. I wonder how you can control for the severity of the psoriasis.
Yeah, so that's not a variable recorded in Trinetics, but you would assume the patients going on biologic therapy would have more severe disease. And we know psoriasis, the more severe it is, the higher risk you are of having cardiovascular events. But in fact, we're seeing the opposite, where they have a reduction in those thrombotic events.
Okay, thanks.
Nice work. Am I right in understanding that you focus the analysis on patients staying on the same drug for 5 years? Single agent?
So one of the weaknesses is we can't confirm patient compliance, and we don't know how long they were on the drug, but we know that they were started on it within 1 year of their diagnosis.
Okay, fair enough. But then are you concerned with the fact that you might, because then the question is, why 5 years, depending on the mechanistic model, because you could have a trigger effect. It's based on immunocardiological schemes for at least arterial thrombotic events. That's one. I don't want to be too long, but that's my concern in your analysis. And how many patients did you exclude who stayed less than 5 years and who might have been stopped from any drug because of a serious adverse event, including a thrombosis or at least MI or something? I mean, that's my... a referee might look into it.
Yeah.
Hi, Nicole Ward, Vanderbilt. So as you're probably aware, there is still some contention about whether or not psoriasis patients are at increased risk for developing cardiovascular disease. Some people believe it, other people don't. When you looked at your data, did you compare your psoriasis patients with non-psoriasis patients to see if the risk was changed in terms of a thrombotic event, in terms of an MI or a stroke?
We did not for this study, but that would be another interesting analysis to perform as well.
Okay. Okay, thank you. Thank you so much. Okay, we'll now continue with the Ryan O'Shaughnessy from Queen Mary University of London, and that will talk about reduced filaggrin expression, reduced ERK signaling, and disrupt desmosomes in atopic eczema. Thank you.
Thank you. I'd like to thank the committee for selecting my abstract. This is a collaborative effort between my lab and Adele O'Toole's lab in Queen Mary and Kevin Mills' mass spectroscopy lab in UCL. And the problem on the left-hand side of this slide is that a major risk factor in eczema is the reduction of expression of filaggrin, either through mutation or just through a reduction in expression. And this inhibits that function that's attributed to filaggrin of keratin filament collapse, and therefore we get barrier disruption. But we wanted to ask the question, are there other mechanisms that lead to barrier disruption in eczema that may or may not be exploitable? So on the right-hand side here, we can see our sort of very sort of small experimental protocol. We have a look at models of eczema by knocking down filaggrin by shRNA, and we interrogate these by phosphoproteomics and by RNA-seq, and then we correlate our findings to eczema samples from the Bangladeshi community in East London as part of the Tower Hamlets eczema assessment. And with these, we have a valuable resource where we can analyze nearly 1,000 tape strip proteomes and do immunofluorescence on a more limited set. And then we put these data sets together to try and determine new mechanisms of barrier disruption that could be druggable in eczema. And to cut a very long story short, we did a lot of phosphoproteomics on our knockdown cells, and we identified our top 10 down-regulated kinases, and 4 of these were MAP kinase related in these boxes here. On the right-hand side is our the results of looking for BMP and TGF-beta related gene expression in our shRNA knockdown keratinocytes, and we could see that there were increases and decreases in expression of BMP and TGF-beta signaling. Oh, that's wrong. Let's try again. There we go. So we looked at our eczema samples to see if what we were seeing in our models reflected the real patients. And you can see in the top panel there, we can see an increase in fluorescence of ID1, which is a well-known BMP SMAD158 target gene. And we see it's increased in filaggrin heterozygotes and compound heterozygotes. And we could show this downstream activation of BMP signaling by immunofluorescence. And by Western blots in tape strips, we could see that our compound heterozygotes had a significantly increased amount of phosphorylated SMAD. Phosphorylated SMAD is inversely correlated with filaggrin overall and positively correlated with keratin too. So it suggested there may be some kind of rescue afforded by having increased BMP signaling in these eczema samples. And to test this further, we treated keratinocytes with recombinant BMP6, and we could see a dose-dependent increase in the expression of filaggrin monomer in human keratinocytes. And this was concomitant with an increase in keratin-1. Conversely, if we treated with a BMP receptor inhibitor called DMH1, we could see a dose-dependent decrease in filaggrin expression, processing, and keratin-1 expression. The other thing we saw, if you remember, was the decrease in ERK MAP kinase signaling, and we could confirm this in our shRNA knockdowns. We had 3 different biological replicates, 3 different knockdowns, and we could see a reduction in phospho-ERK1/2, and we could see that reflected in panel B in our eczema samples. Wild types with high levels of filaggrin had high levels of phospho-ERK1/2, and we could see as the genotype becomes heterozygote to compound heterozygote, we get a reduction in phospho-ERK1/2. And we could see a correlation between filaggrin and phospho-ERK in our samples. We treated panel C as a treatment with BMP of normal human keratinocytes, and we can see that BMP treatment was activating one of these MAP kinases, P38. So we also looked at the global differences in phosphoproteins in our phosphoproteomics of our shRNA knockdowns, and only one group of proteins was significantly downregulated throughout all of our phosphoproteins, and that was very interestingly desmosomal proteins, so this clamp that holds keratinocytes together in the epidermis. And we could see a large number of these genes indicated by the proteins, rather, indicated by the blue circles here, where these phosphoproteins were downregulated. And what was interesting is when we looked at our RNA-seq data, we could see that actually changes in desmosomal protein expression, desmosomal gene expression, was quite modest. However, by Western blot, we could see downregulation of various components of the desmosome in our filaggrin knockdown keratinocytes. Finally, a student in the lab, Kosuma Raman, who visited for 6 weeks and managed to do all of this bottom half of this slide, showed that 2 components of the desmosome, desmoplakin 1 and 2, and desmocollin 3, were mislocalized in alpha-lagrin knockdown keratinocytes. So we can see here cell surface staining in our desmoplakin stain and our desmocollin 3 stain, which is lost in alpha-lagrin knockdown keratinocytes. So how do we put all this together? And this is my sort of conclusion slide. We found by proteomics rather of the tape strip samples that we had in abundance, thanks to the Tower-Hamlets eczema assessment, we could show that a subset of these had high levels of phospho-SMAD158 staining or 158 activity. And this subset of high SMAD158 patients also had high levels of, or higher levels of a number of key desmosomal components. So where we're currently at now is looking to see if there are biomarkers of this increase in desmosomes and whether identifying other proteins that correlate with BMP signaling, i.e. high levels of SMAD158 phosphorylation, comprise genes involved in barrier rescue. And this network here are the 20-plus proteins that we saw correlating to high SMAD158 levels. And in red here are our key proteins that were in kinases and BMP6 and their potential interactions with these proteins. And we propose that these small subset of proteins could comprise potential therapeutic targets that could activate the BMP signaling pathway in eczema to rescue desmosomal function. So I'll leave it there, and I'm happy to take any questions.
Thank you. Okay, any question? So as I understood, these are possible treatment targets to improve the dysfunction of the epidermis, right? So you...
this is very early stage, but we'd like to think that increasing BMP signaling would enhance desmosomal function and therefore rescue barrier function in a subset of eczema cases. We'll need to do much larger studies.
Of course.
This is a pilot study of only 20 samples, but we have access to nearly 1,000, so we can really look at these biomarkers in more detail and try and nail down the ones that correlate with BMP signaling and may constitute potential therapeutic targets, or at least point to mechanistic targets.
Perfect. Thank you. Thank you. Thank you so much. Okay. So let's continue with Sheer Azirland from King's College London. We'll address the effect of age on the severity of psoriasis. Thank you.
Good afternoon, and thank you for the invitation. It is my pleasure to present our work titled The Effect of Age on the Severity of Psoriasis. This work was supported by the Daniel Turnberg Travel Fellowship Award. Life expectancy in the UK is rising, and it is estimated that by 2066, 26% of the UK population will be aged 65 and older. However, research on how psoriasis progresses with age is relatively limited. Our study aimed to investigate the natural course of psoriasis with age in the older population. To achieve this, we conducted an online survey among members of the UK Psoriasis Association, aged 65 and older, who had developed psoriasis before the age of 40. Participants rated the severity of their psoriasis on a scale of 0 to 10 and provided information on lifestyle habits, treatments, medications, and associated medical conditions at various life stages, in 20 years intervals from birth till the age of 60, and 5-year intervals from the age of 61 and above to increase sensitivity within the older age group. The survey also included open-ended questions to explore changes in the morphology and distribution of psoriasis over time. A total of 180 participants were included in the final analysis. The median age was 73 years. 56% were male and 44% female. The mean self-rated severity of psoriasis decreased with age, reaching its lowest point in individuals over 80 years old when compared to severity at ages 21 to 40. This reduction remained highly significant across all age categories above 60. Analysis of qualitative data revealed three main themes regarding changes in psoriasis with age. First, the severity of psoriasis is reduced over time, characterized by diminished redness, smaller and thinner plaques, reduced scaliness, and itch, which aligns with our quantitative findings. Second, psoriasis distribution changes over time, from widespread to localized disease, with increased involvement of the trunk and decreased involvement of the scalp. This change was sometimes associated with changes in hair growth and may be linked to changes in the hair follicle psoriasis axis. Finally, retirement was identified as an age-related ameliorating factor for psoriasis, likely due to reduced stress levels. In search of predictors of high disease severity at an older age, current severity ratings were categorized as high, meaning ratings of 5 and above, and low. Lower disease severity at an older age was associated with the use of statins, as was found in previous studies. This was also associated with longer disease duration. In contrast, the presence of itch correlated with a higher disease severity at any point during the course of the disease. Interestingly, a history of smoking was associated with lower current severity, though the true impact of this could not be assessed due to the low number of current smokers, and is likely attributed to smoking cessation. Our study's limitations stem from its methodology, including recall and selection bias. In conclusion, these findings indicate that the clinical severity of psoriasis improves with age, particularly after the age of 60, and psoriasis morphology changes over time. This improvement was associated with longer disease duration and may be attributed to the aging of the immune system, immunosenescence. In clinical practice, modifications to systemic treatment regimens in this age group are worth exploring on a case-by-case basis. I would like to thank my colleagues and collaborators on this work, and thank you for your kind attention.
Thank you. Any question?
Stina Callis-Duffin found, I don't know, maybe 15 years ago, that the same thing with smoking. And they never reported it because you don't report that smoking will decrease the severity of psoriasis, but her attitude was it's probably because they're less stressed out.
Thank you.
Nick Reynolds, very nice study. Did you look at BMI?
Yes. BMI was calculated as one of the cofactors in all of the analysis, and we didn't find any significant association, but it is worth mentioning that our median BMI was 25, which is relatively low for psoriasis patients. So it's one of the limitations of our study group.
Yeah, the reason I ask is that as people get older, they tend to get thinner, so it may be a factor.
Thank you.
Question.
Yes.
Any indication that the composition or the distribution of immunocytes in lesions of young psoriatics and old psoriatics is different?
There are some studies on the immunologic phenotypes of older versus younger psoriasis, but I think there are various confounding factors. Mostly the age of onset is very influential on the immunologic phenotype of psoriasis. If it starts at an older age, this is not the population we tested. We tested type 1 psoriasis, meaning before the age of 40, but there are studies on this topic, not ours though.
Hi, sorry, William Kings. I have just one question. It was very good to see that you have asked about the determinants of aging, but I was wondering, have you looked at certain markers like genetic expression of psoriasis, especially with patients over 60, or whether you have catered for the underlying differences, the nonlinear decrease in skin genetics when at the peak of 40s and 60s. As one of the paper by Snyder et al. In 2024, just recently, it showed that there's a nonlinear decrease in the genetic expression. So I was wondering, do you cater for that to look at people over 60s? Because it's quite interesting to see that psoriasis is actually decreasing in severity.
Thank you for the comment. It is very interesting. We did not look at this in this study, but it is worth exploring in future studies, the impacts of this. Thank you.
Okay, thank you. Thank you so much.
Thank you very much.
Okay. We'll continue with Monica Molnarova from Scarlet Red Holding from Vienna. That will talk about proof of concept studies, smart imaging, and deep learning for objective psoriasis lesion scoring.
Hello, and thank you for the opportunity to present my abstract. In Scarlet Red, we conducted a proof of concept study focusing on smart imaging and deep learning, and aiming to enhance the objectivity of psoriasis assessment. In cooperation with the Center for Human Drug Research, we were able to collect images from clinical study from 27 patients with psoriasis. The imaging setup was designed in a 2-week fashion, where medical experts took images, full body images, and target lesion images from the patients. Besides of that, the patients were asked to take daily images at home. The images were taken using the Scarlet Red mobile app and standardized via the Scarlet Red skin patch. In total, 358 images were used for the development of the machine learning algorithms. We developed a lesion segmentation and tissue classification algorithm, both yielding accuracy over 85%. Furthermore, we developed an automatic scoring algorithm where, once comparing the results with the expert scores, we achieved correlation of 65, 72, and 85%, respectively, for erythema scaling and induration. Consequently, we applied the algorithm to subsequent images from the same patient in 5 different time points. And as you can see on the plot on the right, we can see a decrease in the scaling component during the time, while unveiling the erythema area. However, when comparing the erythema intensity, we can see a sudden decrease, showing the effectiveness of the treatment. In the last step, we translated the algorithm to full body assessment, where we included automatic body segmentation as a pre-processing step. This step allowed us not only to analyze the lesion composition of the psoriasis, but also localize the lesion on the body and measure the affected body surface area. And with that, demonstrating successful AI assessment of the psoriasis. Thank you for the opportunity to present my work. and I'm happy to answer any of your questions.
We have a question, please.
Hi, sorry, William Kings. Just want to ask, what is the algorithm or the network that you're using in Scarlet Red?
I'm sorry, but that lies under disclosure, so I cannot disclose the algorithm. But we will have a publication soon, so maybe there you can read it.
Okay, that would be fine. And then what about the distribution of patients within your group? Because you said 27 and they're all Caucasian, is it?
Sorry?
Are they all Caucasian patients in the 27
Yes.
participants in your algorithm?
Unfortunately, the study was conducted in Netherlands, and there were no patients included with darker skin type than Fitzpatrick-4, so we don't have currently data. But the concept should work also on the darker skin, but currently we don't have validation data for those done.
It would be very interesting. As well as on this slide, do you have the correlation coefficient of your AI and the expert PARS yet? Because what we want to see is whether the AI is actually correlating to what the expert scores. And the thing is, it's quite hard to see whether it's actually better than the expert scores or not. Just wondering if you have something like that there.
What we observed during our study was that erythema tend to be the most subjectively scored parameter among the physicians. That's why the correlation shows lower in our study. So we need to. This will be probably the next step in our study.
Anyways, thank you.
Yeah, Nick Reynolds, Newcastle. I just wonder how your algorithm calculates induration from a 2D image.
Induration can be calculated during correlation, and also there are already algorithms to measure the depth of the image, and that way we can measure the induration.
Okay.
How does it work on skin of color?
Not yet. For the psoriasis, no.
What about mice?
No, we don't have data from mice either for psoriasis.
Do you think that you could use it for mice? But probably only white mice, it looks like.
But the next step will be analysis on different skin types, as it might bring interesting results.
Okay, so thank you. Thank you so much. Okay. All right, so we'll continue and we'll go to our final presentation by Zoet Dajuve from University of Debrecen, and to present distinct TRM cells characterize psoriasis and atopic dermatitis. Thank you.
Thank you for the kind introduction. So, we all know that atopic dermatitis, AD, and psoriasis are two of the most prevalent immune-mediated skin diseases, and actually we all know their characteristic cytokine profiles, and they share the potential for chronicity. And recent literature data also showed that memory T cells, especially TRM cells, resident memory T cells, have a crucial pathogenic role in both diseases. So in the current study, our aim was to investigate whether these memory T cell populations has different features beyond their cytokine profiles. To answer this question, we used the publicly available CiteSeq dataset of Tsoi et al. This is a 2-year-old, so a really recent dataset, which was published in Science Immunology. CiteSeq, you may know that CiteSeq means that it's a whole transcriptomic single-cell data with some epitope data, so about 40 cell surface molecules. Epitope data is also available for all cells. And this dataset contained 7 samples from skin samples, most importantly skin samples from healthy control, AD and psoriatic skin. And this dataset contains CD45 cells, CD45 positive cells. And what we did, we reanalyzed this dataset. My time is short, so I don't want to go into details, but our point was to create 4 clusters, so we made a subclustering. So we divided based on epitope data, so most importantly based on protein data, we divided all the cells into CD4 and CD8 population, and CD69 high and CD69 low population. So at the end, we had 4 populations. So on the left side, you can see the CD4 cells. In the upper picture, you can see the UMAP of the CD69 high population, which represent the TRM cells, so the resident memory T cells, and the lower population represents other memory cell types, such as Tregs in the CD4 population, an effector memory, migratory memory, and central memory T cells, and other populations. And on the right side, you can see, with this analogy, the CD8 cells. And what you can see in the tables, and I want to highlight that, in both the CD4 and CD8 populations, resident memory T cells were highly, their ratio was highly increased in both populations. And in addition, it's interesting, but we all know that the Treg population was highly increased in the CD4 populations in both diseases. As the next step, what we wanted to do to know which subsets are responsible for the cytokine production. We all know that which cytokines are the major pathogenic cytokines in AD and psoriasis. So, but we wanted to know which subsets are responsible for this, and maybe you can see on this UMAP. So we projected the cytokine positive cells on the UMAP, and on the upper you can see the resident memory T cells, and lower the other population. And you can see that the majority of cytokine positive cells are here, and you can see the color code here. And in AD, as expected, we see IL-22 and IL-13. In the psoriatic samples, we see a lot of IL-17F, IL-17A, and interferon gamma. And which is really important, what we can see that in both AD and psoriasis, the majority of cytokine-expressing cells are resident memory T cells. So as you can see, almost 90% of the cells are TRM cells in both diseases. But it's really important to mention that the clusters within the TRM population are different, which are responsible for the cytokine production in the two diseases. So not the same clusters produce the cytokines within the TRM subsets in the two diseases. And actually, this is true for the, I'm sorry for the mistake, it's not CD4, these are the CD8 positive cells. So the results are really similar. The majority of cytokine expressing cells are also resident memory T cells in AD and psoriasis, but we can see a much more heterogeneous population, as it's only about 50% of the sources of the cytokine produced. So the cytokine source is only about 50%, which was resident memory T cells, and 50% of the cytokines derived from other cells. However, in psoriasis, we can see the same picture, same pattern, that almost all cells, all cytokines are produced by TRM cells. And at the end, we have chosen those clusters within the TRM population in both diseases, which was characteristic for the cytokine production. And these heat maps represent the molecules of our interest. And it's, as expected, we can see that we could confirm the cytokine and cytokine receptor pattern within both diseases. So we can see the so the Th2 pattern in AD, and the Th1, Th17 in psoriasis. But what is, I think it's really important, that when we investigated those genes related to migration or mobility, migration, retention, and homing, what we saw that there is a huge difference between the two skin diseases, and it seems like all those molecules are upregulated in AD compared with psoriasis, which is related to mobility. So it's like AD TRM cells have a higher capacity for migration. They are much more mobile. And that was the same in CD8 populations, where this is the AD cluster. In the AD clusters, we can see those genes such as S1PR1, which is the CD69 antagonist, or KLF2, or L-selectin, which are rather responsible for the migratory capacity of these cells. So if I want to summarize our findings briefly, what we could see is that in both skin diseases, the resident memory T cell population is highly increased compared with the healthy controls, and these cell population is the primary source of pathogenic cytokine in the two diseases. And as expected, we could detect the distinct disease-specific cytokine profiles in both diseases, but most importantly, and which is, I think, is really new, that these TRM cells in AD display a significantly higher mobility or capacity for migration based on these data. And actually, what is our hypothesis is that these TRM cells, what we could see with higher mobility in AD, may be X-TRM cells, which is known from the literature, because these X-TRM cells are formed in the skin, and then they can re-enter to the bloodstream. And actually, this is in line with the fact that in AD we can see a lot of infection compared to psoriasis, where infections are really rare or almost no infections. And maybe this is in line with the fact that now psoriasis is considered as an inside-out AD rather than outside-in skin disease. And thank you for your...
Thank you.
Thank you. If you have any questions, I will
Any
try to
question?
answer them.
Okay, so this is it's an important topic, of course. We have more data on the impact of therapies on TRMs in psoriasis than in atopic dermatitis. So, of course, this is important to better understand the importance of these cells in both diseases and recurrence of disease. Okay. Oh, there's a question. Sorry.
Thank you. Very nice talk. Özge Uluçkan from Barcelona, Almiral. I'm wondering, is just one marker, CD69, sufficient to identify these cells? Are there any other markers when you look unbiased that you could help, look at subtypes of these cells?
It's a great question. You're absolutely right that maybe 69 is not enough alone. Actually, we checked out CD103 as well and other markers, and what I can tell you that in both diseases, the sources of cytokines are positive, CD103 positive cells. So we checked them out. It was hard to decide how to find these, how to differentiate these clusters and how to do this subclustering. But actually, in psoriasis, we could confirm that beyond CD103, that these cytokine-producing cells are CD103 positive, and CD42L positive cells are the interferon gamma positive cells, and low cells are the IL-17A positive cells. So we can see this pattern within the data set. So I think it's true.
Fantastic. Thank you.
Hi, nice talk. I have a quick question. How about CLA marker? Is it there?
A really good question. We need to check out them, but we didn't check out them yet.
Okay, thank you. Thank you. So we'll continue, Johan, with you.
Thanks.
Okay, we're going to move on to the next part of our symposia with our invited speakers. We're running a little bit behind. I think we're about 15 minutes behind so I'll try to catch up a little bit. But I'm going to invite our next speaker to come up, Dr. Marjolein de Bruin-Weller. Please come up on the stage. So I'm going to do a brief introduction. Dr. de Bruin-Weller is a full professor and head of the National Expertise Center of Atopic Dermatitis in the Department of Dermatology and Allergology at the University Medical Center Utrecht in Utrecht, the Netherlands. Her major interest is clinical and translational research in atopic dermatitis. She is the principal investigator in several clinical trials and investigator-initiated studies focused on difficult-to-treat atopic dermatitis and systemic immunosuppressive and immunomodulating treatments. Please read more about Dr. de Bruin-Weller in your program guides if needed. And the title of her talk is Using Biomarkers to Inform the Treatment of Atopic Dermatitis. So Dr. Bruin-Weller, please take it over here.
Thank you for the introduction. And I also want to thank the organizers of this symposium, giving me the opportunity to talk about biomarkers in atopic dermatitis, and especially how it can help us in our clinic. Here you see my disclosures, the firms that I am collaborating with in the field of atopic dermatitis. Now, just a short introduction. We have already learned a lot about atopic dermatitis. It's the most common chronic inflammatory skin disease, characterized by itch, of course, and a severe impact of quality of life. And of course, you have a lot of new treatments available since 2018, but still you see a subgroup of severe patients who is still in need of new treatments because they cannot control it at this moment. A lot of biomarker research have been done in atopic dermatitis, and this is just a broad overview. And in my talk, I want to focus on the predictive biomarkers, so biomarkers that you can measure at baseline that can inform you on the results of your treatment. On the severity biomarkers, because we all like to do objective scoring of our disease, and we know that the clinical scores are not so objective, and maybe biomarkers are. And if you look at the different sources, I will focus on blood, because for the daily practice, blood is the most easy accessible source that we can have, and you can also use it for repeated testing. My first part, I will focus on the prediction of response that we all want to do. And most of these studies are focused on the T2 targeting biologicals. This was one of the earliest studies. It was a phase 2b tralokinumab study. Tralokinumab is an IL-13 inhibitor. And in this study, you see on the left side the intention to treat population. It's the placebo group compared with 3 different doses of tralokinumab. And the figure B and C represent subgroups with high levels of serum DPP-4 and periostin. And those biomarkers were interesting because they are biomarkers for IL-13 activity. And in this phase 2b study, the patient who had these high levels performed the best on tralokinumab. But unfortunately, this was not confirmed in the later tralokinumab program, so you don't see a lot of these biomarkers back now in literature. This is a more recent study of the Japanese group, and they explored biomarkers to predict the clinical effect in dupilumab-treated patients. There were 110 patients coming from several medical faculties. They had moderate to severe atopic dermatitis, and they measured 19 biomarkers at baseline. And the primary endpoint was the association between those baseline biomarkers and the percentage change in EASI score at 16 weeks. And they also looked at the association between those biomarkers and the percentage change in patient-reported outcomes. And this table shows the results. On the left side, the EASI score, and there were no predictive biomarkers found for the percentage decrease in EASI score. But if you look at the patient-reported outcomes, that is the POEM of the patient-oriented eczema measure and the pruritus NRS score, you see that LDH, soluble IL-2 receptor, and TARC were positively associated with the response to treatment. So maybe you can, there is some prediction on the patient-reported outcomes. Maybe a more sophisticated way to use biomarkers is not to focus on one biomarker, but focus on clusters of biomarkers. And here we go back to a publication 2017 from our group, in which 200 patients were studied. Large panel of biomarkers were measured. That was followed by a principal component analysis, and in this way 4 clusters of biomarkers were found that represent 4 different endotypes. And 2 of those clusters had really high Th2 biomarkers. Some years later, we could validate these clusters in another cohort of severe AD patients, and we see that about 48% of the patients are in those Th2 high clusters. And then it would be logical to think that if patients are treated with dupilumab, that especially the patients in those clusters would react very good to dupilumab. And that's what we recently tried to study. It was a study that we've done with colleagues from TREE Germany. So we want to assess the role of the baseline proteins to associate with the response to dupilumab. But I think more interesting, we want to investigate whether those clusters, those endotypes, are related to a better response to dupilumab. So patients from the Dutch Biode Registry and the German TREAT Registry were included. Patients were treated with a normal dose of dupilumab, and at 16 weeks the EASI score was done, and the patients were then divided in 3 response groups. Patients who could not reach EASI 50 or 50% reduction of EASI, they were the low responders. The intermediate responders, EASI 75, and then the high responders, the EASI 90 or more. We measured 16 proteins, and that panel was based on our previous studies with endotypes and on disease severity. The baseline protein concentrations were compared between the different response groups, and random forest models trained on the baseline data were used to whether the response could be predicted on those baseline biomarkers. And then again, we defined the clusters based on the principal component analysis. We could include 127 patients with a mean EASI of 19, so moderate to severe population. And we had a rather nice distribution between the response groups at week 16. But unfortunately, the random forest models trained to predict the response achieved an accuracy of only 35%. So we could not have the baseline proteins predict our response in our cohort. But we again found the 2 clusters, T2 dominant and non-dominant clusters. And that's what you see here on the left side. The blue are the TH dominant patients, and the orange one is the TH non-dominant cluster. And we could find 35 proteins that were significantly different between those clusters. But when you look at the response groups, you see that there's a random distribution of the response groups between the 2 clusters. So although everybody thought that maybe those 2 Th2 high patients would react the best, we could not demonstrate that in our study. More or less the same was done in a Chinese population. Here 125 patients were studied and 24 cytokines were measured. Here also 2 clusters were found. They were not completely the same as our clusters, but there was some overlap. But also here there was no association with the efficacy of dupilumab. So in summary, for this first part, you see that most of the prediction studies focus on the Th2 targeting biologicals, and especially dupilumab. The prediction based on single biomarkers does not work. But unfortunately, also the prediction of the response based on the endotypes from which we expected more was not successful. But there are some problems with these prediction studies, and that is especially how you define your response. We choose now for relative values at week 16, and maybe it's better to choose for absolute values for disease control, or should you use physician outcomes, or should you use patient-reported outcomes, or maybe a combination. And that can all affect your results. And also the time point that you choose for your response. Because we found out that a lot of patients who were not reaching EASI 75 or 90 at 16 weeks did reach it at week 28. So they are more late responders. So we don't see a lot of really non-responders. And that makes it so difficult to do these prediction studies. Then the second part of the study, can we have an objective measurement of disease severity in AD when you use biomarkers? I first want to show you this systematic review and meta-analysis. And here you see some cross-sectional studies who measured several serum biomarkers and related that to AD activity. And here several clinical scores were used. It's not only EASI or SCORAD, but also some older scores. But what you can see that the most research has been done on TARC, serum TARC, and you see that overall the correlation is rather high. And that's also for the related biomarkers MDC and CTEC. But also IL-18 seems to perform very well, and LDH a little bit less. And interesting, a lot of studies focus always on IgE. It started already 50 years ago. But overall, you see that the correlation between disease severity and IgE is not very, very good. And that's also true for ECP, CD30, and vitamin D. Here again some data on the Japanese study, which I already showed you some data for the prediction. What they also did was measure the biomarkers and related to the scores at baseline. So you see on the left side the EASI score, and then related to the 19 biomarkers that they measured. And you see then a lot of significant associations. And they also did this for the patient-oriented eczema measure and for the pruritus NRS. And then on the right side, you see that in the middle of all the circles, you see LDH, eosinophils, soluble IL-2 receptor alpha, and TARC, who performed well in all those three outcome measures. So they seem to be the best biomarkers for this. When you want to evaluate your treatment, it's of course important that the biomarker performs very well in longitudinal studies. And if we then go to the meta-analysis, we see that only TARC seems to perform well in the longitudinal studies. And I also want to show you some biomarker results from our Dutch Bioday registry. This is a treatment with dupilumab. You see a nice change in EASI score on the left side. And if you then look at the severity biomarkers, that's especially TARC, PARC, and periostin, you see a nice decrease over time. Also IL-20 performs rather well. And we also looked at eotaxins, because eotaxins are responsible for attracting the eosinophils into the skin. And also there you see a nice decrease during treatment with dupilumab. If you want to improve your objective measurement, maybe it's better not to measure one biomarker because the disease is so heterogeneous. Maybe combine some biomarkers. And these are studies are done by Daphne Bakker and Judith Thijs from our department. They measured a lot of biomarkers during dupilumab treatment, and they found out that TARC, IL-22, and soluble IL-2 receptor, in combination with a very complicated formula, that resulted in a nice prediction of the EASI. So on the right side, you see in blue the EASI that was measured, and you see in green the predicted EASI. And we also used this model for cyclosporin for topical treatment and for tralokinumab, and it performed very well in all those treatments. I already told you in the beginning that blood is easy accessible and maybe the best source for biomarker use in daily practice. But there's also another way that you can use blood samples, and that is by dry blood spots. So it's just a simple finger prick. You can put the droplet on the filter plate. You can dry it and you can store it, or a patient can do it at home and send it to your office. And later on you can elute it and then measure the biomarker levels. And if you look here in this study, you see correlations between EASI and serum biomarkers, but also EASI with dry blood spot biomarkers. And then you see that especially TARC performs very well, even better than in serum. So this can be a possibility to do at home. It's less invasive than a venipuncture. It's not painless, but it's more easy to do. So, summary regarding our objective disease measurement with the biomarkers. If you look at cross-sectional studies, you see several biomarkers that are correlated with EASI, with the strongest evidence for TARC, CTEC, MDC, and IL-18. But we see also evidence for LDH and soluble IL-2 receptor. If you want longitudinal follow-up, we see that TARC performs the best. There are studies with topical treatments, the oral immunosuppressants, and the T2 targeting biologicals who show that TARC is a very nice biomarker for severity. You can improve the correlation with the EZ if you use a panel with biomarkers. And you can also use the dry blood spot if you want to do it less invasive and maybe for home monitoring. So when we come back to the title of the presentation, using the biomarkers to inform your treatment, you can conclude from this that TARC is still the best option to monitor your effect. But we experienced a problem last year because we started treating patients with JAK inhibitors. And here you see 3 examples of patients who were treated with baricitinib, upadacitinib, and abrocitinib. In red, you see the EZ go down very nicely. But if you look at the blue line, the TARC, it's high, it stays high, and even increases during treatment. So this is not what we expected. So TARC seems to be the ideal biomarker, but not anymore. And of course, it's also interesting, what does this mean, this increase in TARC? So we did some further research, but I will not tell about this because it will be presented tomorrow afternoon by Lian van der Gang, and it's also on poster 77. So if you're interested, go tomorrow to Lian. And I want to thank the clinical and translational AD group of the University of Utrecht. Thank you.
Well, we have time for 1 or 2 questions. Anybody in the audience have any questions?
Yeah, great talk, and I think the emphasis on replication in multiple cohorts is crucial for these biomarker studies. Yeah. I remember from your early work there was an association with filaggrin status as well, but that seems to have been dropped.
Yeah.
Is that right?
Yeah. Yeah.
That doesn't help you?
Yeah, we try to look in our cohort to the filaggrin mutations, but it doesn't matter where you have a mutation or not, homozygote, heterozygote, it doesn't matter.
And just a suggestion that in machine learning, you're probably better to use the continuous variable rather than this categorical because it will improve your outcome likelihood.
Yeah, thank you.
Yeah, I got a question. Great talk. In terms of the biomarkers, it seems to be quite noisy and a lot of heterogeneity in the ones that you've been using. Are we looking in the wrong place, or are we using a too small of a panel, like the O-link has up to now 3,000 proteins that it can measure at the same time? Or do we need to incorporate other types of omic data to get better? What do you think?
Yeah, and of course there's a lot of debate whether you should measure in the blood or in the skin, and maybe combine blood with skin, maybe skin strips, but it's far too complicated in the clinic. So I think for the discovery, O-link is nice, and there are some studies running, and there are some data from O-link, but also there you don't see the holy grail. Yeah. And I think the most remarkable thing is that now with the JAK inhibitors, our ideal biomarker doesn't work. So it also depends on the treatment. And that was new for us.
Yeah, complicated, right?
Yeah.
Well, thank you. Well, I'd like to invite our next speaker to come up, Dr. Satvir Mahil. She is a consultant dermatologist at St. John's Institute of Dermatology, Guy's and St. Thomas NHS Foundation Trust in London, UK. She's an adjunct senior lecturer at King's College London, also in the UK. She has a major clinical and research interest in psoriasis, and she is the co-director of the So Protect and So Protect Me global registries. As director of St. John's Derm Academy, she leads the development and delivery of dermatology educational resources for healthcare professionals. I'm delighted to have you up here, and the title of your talk is Using Biomarkers to Inform the Treatment of Psoriasis.
Thanks so much. So it's a real pleasure to be here and very grateful for the invitation to talk today. Let's see. Oh, sorry. Oh, do I have to click the talk? Sorry. Okay, fantastic. So pleasure to talk about biomarkers to inform the treatment of psoriasis. So precision medicine is powered by data and enabled by biomarkers and a better understanding of disease mechanisms. It aims to tailor disease prevention and treatment approaches by accounting for these inter-individual differences that we've been talking about in genetic, molecular, environmental, and lifestyle factors. And we can think about precision medicine in the context of trying to prevent disease onset and progression, enable early targeted treatment, and ultimately try and improve long-term prognosis for our patients, so reduce their disease and healthcare burden over their lifetime. And the hope is that we can move from a one-size-fits-all treatment strategies to more individualized treatment. So perhaps something like this might be a vision for the future. So wouldn't it be great if, when somebody presents with new onset psoriasis, we could analyze their predictive biomarkers, and as we're saying, potentially it would be a series of biomarkers within a prediction tool in order to assess their risk of developing severe disease over their lifetime. This might then enable us to early on in their disease course allocate the right treatment. So, for example, if they have a risk of severe psoriasis and psoriatic arthritis, we might allocate a particular medication. If they were at risk of severe psoriasis and no psoriatic arthritis, perhaps a different medicine. And if they had low-risk psoriasis over their lifetime, perhaps the emphasis should be more on providing education, empowering them to self-monitor and self-manage. And in the end, what we want to try and achieve is for those who are highest need and who have risk of most severe disease to induce remission or skin clearance as early as possible, because this might help ultimately for them to stop medications later on in their life and potentially achieve drug-free remission or cure. So with that vision in mind, how are we doing? Well, the good news is, and as we've heard today, we've got fantastic treatments for psoriasis, and we've got a plethora of biologic agents that we can use in our therapeutic armamentarium for people with moderate to severe disease. As you can see from this network meta-analysis plot, this hierarchical cluster analysis on the right there, back in 2020, even then, the biologics are clustering towards the right side of the plot, so with higher efficacy. So this was a plot of data from more than 30,000 patients and 62 clinical trials. Fast forward just a couple more years, and this is data from the Cochrane database of systematic review. So this is a living network meta-analysis. And the plot's more dense, there are more agents, but again, they're clustering towards the right, so high efficacy. But that was data from clinical trials and from the induction period of treatment. But what we are really interested in increasingly is looking beyond that period into the longer term, and of course, how does this relate to the patients that we see in our practice? So when we think about durability of biologics in real-world practice, we can look to pharmacovigilance registries to help us with data. This is from the UK pharmacovigilance registry called BADBIR, more than 16,000 treatment courses, and you can see that there is variation in durability of response, and the highest survival appears to be with the IL-23 inhibitor. You can see gozolkimab there, but this paper teased out some of the potential effect modifiers and highlighted the presence of psoriatic arthritis being important, previous biologic exposure, and ethnicity, as we've heard. So we're interested in trying to find molecular genetic biomarkers, and there have been long-standing and large-scale efforts towards exactly this goal. This is a chart that's come from work by the BioMap Consortium, which is an academia industry consortium across Europe for both eczema and psoriasis, although this focuses on candidate biomarkers of treatment response in psoriasis. There's been a lot of activity, as you can see, and one of the key take-home messages is that mechanistic biomarkers are more likely to be informative and robust. And these are biomarkers that are involved in disease pathogenesis or drug mechanisms of action. I've just highlighted T17 cell differentiation and activation and positive regulation of NF-kappa B as categories that some of the biomarkers fall into. But a very important message is that no biomarkers were supported by sufficient evidence for clinical use without further validation studies. So another piece of work from Biomap was there to remind us of the important criteria that we need to tick off for a high-quality biomarker. And it ranges from some of the themes that we've been talking about today. So not only should the biomarker be sensitive and specific and reliable, but it should be feasible to actually for us to use and roll out in day-to-day practice. So the assay should be minimally invasive, easy to perform, have a short turnaround time. They should be cost-effective, of course, and that independent validation is key. So there are a number of steps that a biomarker has to go through, and it's a long and arduous path, so not many make it through out the other side to implementation. But one potential sort of success story is serum drug levels. And back in 2013, we did a very small-scale, single-center study of a small number of patients, but it showed that serum adalimumab drug levels, as early as 4 weeks into treatment, predicted response at 6 months. This then, a few years later, was validated as part of work that Catherine Smith led and funded by the UK MRC SORT consortium. This also showed then that early serum adalimumab levels predict 6-month responses. And as part of this work, we were able to establish a therapeutic range for adalimumab. This has then been implemented into clinical practice. It's had reach into guidelines and real-world practice. And in protocols that we implement in our clinics, we are doing both reactive therapeutic drug monitoring as well as proactive therapeutic drug monitoring using this range. So beyond serum drug levels, there's been a lot of interest in genomic biomarkers. And we've heard from Sarah about HLA-C0602. So there's been a lot of interest in this as a biomarker. It is the major genetic determinant of psoriasis, and it has a greater genetic effect than all of the other susceptibility loci combined. It encodes an MHC class 1 receptor, as you can see here. And in the context of it being a biomarker, again, work by the SORT consortium showed that individuals who are HLA-C0602 negative have a better chance of a response from adalimumab compared to ustekinumab. Now, this could be an interesting finding in the context of the ustekinumab biosimilar sort of landing in our therapeutic armamentarium very soon. But this data came out in 2018, and it's not yet reached the clinic, and it may be an example of how the pace of change of therapeutics can actually supersede this timeline from biomarker discovery, validation, through to implementation. And when we go back to our network meta-analysis plot, you can see where adalimumab and ustekinumab are sitting, and there are a number of other biologics that are more shifted to the right. So perhaps the decision should be less about which biologic should we select, but more when should we start the treatment. And this goes back to the original sort of vision that I presented at the beginning. So if we think about this prediction tool to assess risk of severe disease, genomic biomarkers may well help us there as well. And this is recent data from our group of, it's an analysis of more than 40,000 individuals across 4 population biobanks. And Catherine will talk about this a bit more shortly, but essentially it shows that polygenic risk scores may help to identify those who are at higher risk of developing severe disease. But also, if you look at the multivariable logistic regression analysis there are other clinical and demographic factors that may well be important, including smoking status, age of onset of psoriasis, and body weight. So perhaps the next step is now to integrate both those genomic biomarkers, the polygenic risk score, with these other clinical factors into a multimodal stratification tool, because this may have better ability to discriminate psoriasis severity than any one of those factors alone. If we are able to intervene early, where could that take us? Well, there's interesting data that's being released from the GUIDE study, which is giving indicators as to the promise of this approach of early intervention. So they've taken a group of patients with psoriasis and treated them with the IL-23 inhibitor, gazolcamab, and they've shown that those patients who have a short disease duration, so less than 2 years, have a higher chance of achieving complete skin clearance than those who have a longer disease duration. And although long disease duration, for the purposes of the study, was more than 2 years, the mean duration in that group was actually 20 years. So there's a better chance of success if you can intervene early, and then the plot on the right there shows that also if you're biologic naive, that can potentially improve the chances. And then when we move forward into the longer term, if your patients achieve skin clearance, then the promise is then that potentially you may be able to reduce their drug dosing, so lessen their drug burden over the longer term. So they took their patients in the GUIDE study who were completely clear, they randomized them at week 28, and then for one group, dosed them 16 weekly with gazolcamab, and then for another 8 weekly as per standard care. And they found that 16 weekly dosing at 68 weeks was non-inferior to 8 weekly dosing for maintaining disease control. So there's potential to reduce the dosing, and there's also hints from other clinical trials that this is possible too. So this is some data from risankizumab clinical trial. Again, patients who were completely clear were taken just 28 weeks into treatment, and when their biologic was stopped, you can see 6 months later, half have actually remained clear. So there's potential to reduce the dosing, and what we're interested and excited about is trying to personalize that dose reduction. And we're investigating 2 strategies. So one is patient-led as needed treatment, and the second is therapeutic drug monitoring guided treatment. And this is part of a new feasibility trial called the Planned Psoriasis trial that we're running, where we are taking a group of patients who have well-controlled psoriasis, and for some, we're advising them to do patient-led as needed treatment. So this is when patients dose themselves at the first sign of their disease coming back. Potentially, this may reduce their long-term drug burden and lower costs. And the second strategy is therapeutic drug monitoring guided treatment. So this is building on some of the work that we've done in adalimumab, but really taking it to the next level, because we are individualizing their dosing based on their serum drug levels and other individual covariates, such as their body weight, serum albumin, and creatinine. And we're doing this through a pharmacokinetic model that's been embedded in a therapeutic drug monitoring dashboard. Charlotte, who's our research fellow, will be talking more about the development and validation of the dashboard tomorrow, and is also presenting poster 79. So there looks to be hopefully potential to reduce the drug burden through dose minimization. But what about this vision of drug-free remission one day? Well, at the moment, what we observe is when we stop our patients' biologics, their disease does ultimately come back. And it comes back in the same place that they had their plaques before. So this is indicating that there's an inflammatory memory that's retained in the skin. And it's very important that we try to find out more about this going forwards. It's likely to be at multiple levels. So inflammatory memory can be at the subcellular layer, so DNA. It can be as part of epigenetic modifications and in RNA and protein. It can be retained within the actual structure of the tissue, and also, of course, as within tissue-resident cells. And we've heard already today about these tissue-resident memory T cells. So there's been a lot of interest in recent years about this population of cells. So they are responsive to IL-23, and they release IL-17, the CD8 positive. And the hypothesis is that if we intervene early, we could actually deplete this population or reduce the abundance of these tissue-resident memory T cells in the skin, such that then when the time comes to stop the treatment, the skin has less of a propensity to have psoriasis recurrence. So that sort of disease modification is it's an exciting concept for the future if we understand more about what the nature of this inflammatory memory is. So hopefully with all of the work that we're all doing collectively, we might be able to achieve this goal, but hopefully something to discuss later. Thank you very much for listening.
We have time for at least a couple of questions. If anybody has it from the audience. If not, I actually have a couple. So in terms of the early intervention, I think that's actually quite a hot topic in the field at the moment. And the GUIDE study had, I think they defined the early onset at the 2-year mark. And then you have the step-in study at, I think, six-month mark. Where do you feel like is the right time to intervene? Should we, is it gutted psoriasis, or do we need to truly need it to be plaque psoriasis before we intervene? And
I mean, it's interesting when you think about the inflammatory memory. I mean, that probably happens. It's starting straight away. So the earlier that we can go, the better. But designing studies like that is incredibly challenging. To find, to really get the patients. It's really about then reaching into primary care or into the community to get the patients. But I think as soon as the disease starts, that seems to be the place that we almost want to investigate straight away.
Yeah, I guess it's a problem when it takes 6 to 9 months to get a dermatology appointment.
Yeah, absolutely.
Yeah.
There's many barriers.
Herbie?
Great talk, Sylvia. Every bachelor's from Paris. I have a question regarding, first, a comment, because you introduced your talk saying which and when. And then you accentuate, you give more emphasis on when, more than which. May I challenge this and try to reintegrate what Michel Gillet and Johan said about the plasticity of the inflammatory circuits. And my question for you is, did you try to investigate the biomarkers at the time of the patients are relapsing without evidence of immunogenicity, without anti-drug antibodies, with, let's say, response-associated plasma trough levels of the drug? Because the question is, are these patients potential switchers, right? And so I think you have a great tool, and this is not necessarily the majority of the patients, but when we discuss this, there might be some patients without immunogenicity, and there should be something that...
No, absolutely. And I guess the point that you're making is you can't consider these things in silos. It has to be this integrative approach where we can look at the serum drug levels, we can look at the transcriptome, and look at the phenotype and try to pull it all together, because the disease is far more complex than just one factor alone. So I guess what the data shows is that there are signals there, but to fully make clinical impact, we'll need to, refine it and bring it all together, definitely.
And that's my last comment, because you'll see Florence Hassan will report on that, some type 1 interferon-driven psoriasis, like true psoriasis. No matter what you are intervening early, if you are not using the right drug, it doesn't work. It might worsen, actually.
Yeah.
Thank you.
Thank you.
One final quick question. So I sometimes hear from my colleagues when I talk to them about biomarkers, is that suddenly say we don't need them anymore. Now we have drugs that give 90% clearance in the great majority of patients. What is your take on that?
I mean, it's a great position to be in, where we have some fantastic therapeutic, but we still see the inter-individual heterogeneity. We see it in clinical practice, and we have the patients who fail and fail again. We have the multiple failures. We have, there's the challenges with comorbidities, which we're going to learn about soon, obese population. They're increasingly difficult to treat. I think it will always be a challenge, but our starting point is great. I think we're at a very good point where, we're ahead of many other common diseases. However, the challenges are still there.
Great.
Yeah, enough to keep us busy.
Well, thank you. Agree. Well, we'll invite our next speaker up, Dr. Christian Westergaard. So Dr. Christian Westergaard is a clinical chair professor at Aarhus University Hospital's Department of Dermatology in Aarhus, Denmark. His research focuses on the inflammatory reaction in the skin and its influence on skin barrier function, but also on the epidemiology of comorbidities in atopic dermatitis. His research group also focuses on bullous diseases, mastocytosis, and patient-reported outcome measures. The title of his talk is Using Biomarkers to Assess the Risk of Comorbidities in Atopic Dermatitis. And I'm looking forward to your talk. So
thank you. And thank you very much for the invitation to come here and talk about this exciting subject. It took me on some paths that I had no idea that I was going to go down. So I'm going to talk a little bit about these biomarkers to assess the risk of comorbidities. We've already heard a lot about biomarkers, about different proteins to measure, and I think we've had some wonderful talks until now. These are my conflicts of interest, and I'm going to go straight onto this because I wanted to check out what really is the definition of a biomarker, because we have been talking about biomarker for quite a long time. And I found this definition from the National Institute of Environmental Health Sciences. And what it says is that a biomarker is an objective measure that captures what's happening in a cell or an organism at a given moment. Now, this really opens up for a huge bunch of things that we can measure, right? It's just an objective measure, but it is an objective measure. And so where can we find these biomarkers going down on this? Well, we've talked a lot about this until now, and this is really the classical biomarkers that we find, genes, proteins, lipids. We can measure the microbiome, what goes up, what comes down, and there's a lot of other molecules that we can measure from serum, from skin, and so on. We can also do imaging. We can do CT scans. We can do DEXA scans if we want to look, for example, for osteoporosis. The risk of osteoporosis is something that we get out of the C-score and the T-score from a DEXA scan, right? We can also do physical measurements. We can measure blood pressure. This tells you something about the risk of developing cardiovascular events. We can do spirometry to tell you something about the risk of developing asthma. We can do patch tests for contact allergies. Maybe not a biomarker, but it's at least a diagnostic marker. We can also do functional tests like exercise tests. We can do a glucose test to test you for diabetes, to tell you something about whether you have a risk for developing or if you have a latent diabetes. And this is also something that we talk about when we talk about comorbidities. We can do PROs. This is an objective measurement. It's really a subjective one that you get from the patient, but it's something that we use. We can ask the patients how they feel, how the treatment is working. And finally, and this is the latest, and now I'm really going out on a limb, we can do digital biomarkers. We can look at wearables. We can get data from, like a watch like this. We can look at pulse. We can look at movement. We can do voice analysis to check if patients are getting depressed. There's a lot of things here. We can look at online activity from the patients. So there's a lot of biomarkers that we can start out with. So 20 minutes is not really enough. We've also talked a little bit about this. Diagnostic biomarkers, right? Which disease are we talking about? Like serum ACE and sarcoidosis. We can talk about prognostic. This is something that we've just heard about, something that we're really, really interested in. The outcome of the disease or the severity of a disease, and in this case, what comorbidities are the risk of, and are the patient in a high risk of developing comorbidities. And finally, also the response to treatment, which I find is very, very interesting, and we are in my opinion, as we just heard, at a good starting point, but we are not there yet. So in terms of atopic dermatitis, diagnostic, clinical diagnosis, we can do biopsies and we can do a support from serum IgE. Prognostic is the risk of asthma, contact dermatitis, food allergy, and cardiovascular disease. And finally, also the response to treatment with the JAKs and the biologics and nutritional treatment. As we've just heard, it would be great also in AD if we could do a blood test when they came through the door and say, well, you need to go straight to this treatment rather than trying out a lot of different treatments before we find the right one. But what are the comorbidities in atopic dermatitis? I tried to just put them up here, and we're going to discuss some of them. We have the typical type 2 inflammatory diseases that we see together with AD, like asthma, hay fever, food allergies. Then there's the cardiovascular diseases. I know that this is a matter of debate. I don't think there's any doubt that there is an increased risk of cardiovascular diseases in this patient group, but whether it's due to the atopic dermatitis itself or it's due to the lifestyle, that's a whole other discussion and not one we're going to have here. Then we have psychiatric diseases. We know there's an increased risk of psychiatric diseases in this patient group, but whether it's due to the fact that they're more stigmatized, that they have less social activity, or if it's due to the ongoing inflammation is also a question that still remains to be answered. Contact dermatitis, an infection, and also, again, maybe taking it a little far, the social and educational life of these patients is also something that's affected by the disease and may be counted as, a comorbidity. Now, these are some studies, and I've called it a simple approach to biomarkers in AD, and I'm aware of that. But we wrote the study ourselves, so it's okay that I call it simple. But it's a little bit old, and what you can see here is that we looked at this group together with the group in Kiel, and we looked at 4 different alarmines, and we looked at IgE between, healthy controls and AD in children and in adults, and we also compared the children with the adults. Down here, you can see this is really very simple. This is some of the things that we heard also. It's just one cytokine, and we can see whether it's increased or decreased in these groups. But we also tried to relate it to comorbidities and to look at whether they had allergic rhinitis, allergic asthma, if they had sensitization to dietary allergens or even to inhalant allergens. As you can see, it's a bit here and there. We did bonferroni correction, and we can find a few of them. But what you also see is that quite a large standard deviation. So whether it's diagnostic or not, maybe not, but it's really it's a clue to go for. If you look down here, you can see, for example, here with the inhalant allergens, it's quite increased in the group. If we look at the total number of AD patients, the serum concentration is quite increased here compared to the healthy controls, but not as you can use it for a diagnostic mean. We just heard about all the validation that we need in order to use them like this. And then there are other studies too, like you can have the toll-like receptor mutation together with IgE is associated with asthma, allergic rhinoconjunctivitis, high eosinophils and food allergy, ST2 and IL-33 together with bronchial asthma and cardiovascular disease, IL-23 may, but you see the code word here is may and can be and correlation and perhaps. So it's not really something that we can, it's not a 0 or 1 response that we have here if we just take these ones. And then I'm very happy that Marjolein showed the initial study, because I'll take the number 2 study because this is the figure that shows the same thing. And it's really a very, very impressive study measuring all of these proteins in the blood and having these 4 clusters. I don't remember if it was 130 or 150 different proteins that you measured. It's really amazing. And here you can also see that the clusters that you mentioned, that there is a difference between some of the comorbidities, maybe not a lot, but still some. You can see that cluster B, for example, has a higher risk of, can you see the pointer? Yes, you can. Higher risk of asthma compared to cluster C. And down here you have the rhinitis. So there is some differences in these clusters, also with regard to comorbidities. So we are on the right track, right? But there's still a lot of overlap between the clusters and the comorbidities. Then we have the sleep disorders and poor quality of sleep, because we know that AD patients, they don't sleep well. They have longer latency of sleep. That means that it takes longer for them to fall asleep. They have an increased fragmentation of their sleep. they have difficulties in waking up, so they get a poor efficiency out of sleep. Maybe that's not a nice thing to say about sleep, but it really means that you don't get rested. And we know that this is high risk for ADHD, for obesity, for hypertension. So suddenly we're talking about this is a risk factor for both psychiatric disease, but also for cardiovascular disease. And how can we measure this? Do we have something to measure this kind of sleep quality with? And there's been several different ways to do this. There are PROs. There's an overview here of the different kind of PROs, questionnaires, because we really need to ask the patients with PROs. These are validated. I can tell you something about the quality of sleep. I'm not aware of any studies that takes these PROs and correlates them to the risk of psychiatric disease or cardiovascular diseases in AD patients. You can also do the wearables, right? Like this, just a simple sports watch. You can get it for less than, 50 euros in any sports shop. Very popular. And they can tell you something about both sleep quality but also movement. But you can also do something with a mattress or something called ballistocardiography. Yeah. I'm not sure that I would wear or even sleep with one of these, but it tells you something about the respiratory movements, but also the ECG, and it's something that tells you something about the sleep quality of the patients. Then you can also do the CT scans, and this is also a study that we did in our group some years ago, and what we really did was that we did CT scans of the coronary arteries. So simply enough, we looked for atherosclerosis, and we separated the coronary arteries into 18 different segments and looked at how much atherosclerosis did they have. And what we found was that if you look here, you can see that among AD patients, there was an increased degree of atherosclerosis as counted by the segments that were involved with atherosclerosis in these patients, when they had mild atherosclerosis and moderate atherosclerosis and compared to psoriasis, where psoriasis had the very severe atherosclerosis. And not that I'm going to show you, but this is really a biomarker for the risk of developing cardiovascular diseases in these patients. Again, we didn't have any data on whether they smoked or not, whether this was due to the disease itself, but it told you something about the risk of having cardiovascular events. Finally, the digital biomarkers. There's some work here from Alexander Sink's group, and I would like to, this is not AD, but this is looking at pollen in Germany and Sweden. And up here, you can see in the first graph in A, this is pollen over Germany, and down here in C, it's pollen in Sweden. What you can see here is crowdsourcing, where you go out and you ask Google how many has searched on terms related to asthma on Google in Germany at this point in time, and how many in Sweden. And as you can see, it completely follows pollen season, the real pollen numbers. So it's really something that we can use to detect things that takes place in real life when you do this kind of crowdsourcing. And they did this in AD, where they looked at social media. This is a French study where they went in and looked with text analysis on what was written. I know I'm a little bit out in biomarkers, but it's still a marker. It's something that we can measure objectively, right? And they looked at it, and they could look at controlled AD and uncontrolled AD, and they could see that there were specific words being used in these texts on the social media, depending on whether you controlled your atopic dermatitis or not. And again, also here, you can see search terms on Google. This is again from Alexander Sink's groups, looking in Germany. And what you can see down here is that if you look at comorbidities, there is an increased search here, and this is the number of searches over time. So it does not tell us anything on the individual patient plan, but it tells us something that there is a gap in perhaps our treatment or perhaps our information of these patients in telling them about the risk of disease. But I think it's something that's still in its birth, but it's something that we're going to use a lot more over time, these kind of analyses of what takes place in the digital life. So in conclusion, I think we just heard about the diagnostic biomarkers, but maybe we need some more reliable prognostic and predictive biomarkers. And now you even destroyed TARC, right? So we need something new there. We have a lot of associations with different kind of things, but we need a combination of these. And I think the Dutch study is really showing this is the way forward, but maybe we need to combine it with other things too, like digital behavior like spirometry, like DEXA scans in order to tell us something about the comorbidities. And finally, you know, I think digital biomarkers, maybe that's a bit far-fetched, but I told you it took me down a road where I didn't know I was going. Digital biomarkers could be a path to predicting disease severity. So with that, thank you very much for the invitation to come here and speak.
We have time for some questions. Well, I got one for you. So I found it to be very informative. So there's a lot of things that we can measure and kind of collect information on. But at the same time, the more we measure, the more it's going to affect the overall power of what we can do. You need progressively bigger sample size as the more things you're kind of measuring. How should we go about identifying which biomarkers we actually want to measure and which ones should we prioritize on?
I think this is really a very good question, because now I just put a lot of things to the table, right, that we can measure this or that. I think that we are actually doing it the right way, because like we're meeting here and we're discussing, you know, could you use a wearable to maybe measure sleep quality? And if this turns out to be positive, then it's something that we might want to use together with the results like the ones from Holland. So we have to take it step by step, you know, bite by bite. So it's really difficult to maneuver in this one. But on the other hand, the power that we get now to data process, right, it's growing exponentially. So I think that within a few years we'll get more and more data, and we'll do all of these kind of machine learning, whatever it is, to learn these kind of things. So maybe the problem is solving itself.
Nick Reynolds, just to add to the debate, I mean, I think one thing we haven't discussed actually is that I think in clinical practice, atopic eczema is much more variable day to day than psoriasis, and patients report that. And Maiko Tanaka from Imperial in London has done a nice study where she's got patients to report daily their symptom score on an app, and has used the pattern of development of those symptoms to predict outcome a few days away. But you know, you're picking up flares early, and maybe we have to go back to basics and think about, because I think that happens even when people are on dupilumab, doesn't it? They still go along with little flares. So I think thinking about it as a continuous variable with easy measurements may be a very good way. You know?
I agree. It could be like an app or PRO combined with something in how you sleep, measure your sleep quality, which could then predict, do I need to intensify topical therapy for the next couple of days? I agree completely. Yeah.
Great. Well, thank you. Great talk. I'm going to invite our final speakers of this afternoon up on the stage. So Dr. Catherine Smith, she's a professor of dermatology and therapeutics and a consultant dermatologist at St. John's Institute of Dermatology, King's College London, and Guy's and St. Thomas Hospital. She directs the Skin Therapy Research Unit and is a lead clinician in a national specialized services for adults with severe psoriasis. Dr. Smith's clinical and research interests focus on inflammatory skin diseases and translational medicine, extending from biomarker discovery to interventional studies, and she also has a major interest in guideline development and implementation in the United Kingdom and Europe with the aim of bringing research findings into clinical practice. The title of our talk this afternoon is Using Biomarkers to Assess the Risk of Comorbidities in Psoriasis. Dr. Smith, looking forward to your talk.
Thank you very much. Thank you. I'm aware of the last speaker, so I'm actually in a very good position, because a lot of what I've been said has been said by Christian and the whole speakers that have gone before me. But this was a tricky title. This is a massive subject. I'm glad you found it challenging too. I thought actually I might just start. John Updike, I don't know how many of you have read his books. They're fabulous. But he also had terrible psoriasis from the age of 6. And it changed his life. It's in fact why he was a writer, because he didn't want to show his skin. And he talks and writes very movingly about the stigma and effect on his mood. And of course, mental health problems are a comorbidity, but actually he was otherwise very fit, and he made this comment that people with psoriasis are actually in very good health. And I wonder whether we feel that that's still true today. I mean, epidemiologically, most people with psoriasis have no more or less comorbid a burden than the general population. But nevertheless, there is a burden of comorbid disease that is important and is definitely more prevalent in those with more severe skin disease. This is a lovely study in 2006, just illustrated myocardial infarction. I don't think there is any doubt that there's an association between cardiovascular disease and psoriasis. It's the causal relationship that's in question. But we do know also it contributes to the cumulative life course impairment in people with the disease. So it's important, and therefore there's an opportunity to improve outcomes with biomarkers. So I'm going to talk about some general principles where we are now and looking forward, because actually in reality, apart from clinical biomarkers, which I'll talk about briefly, there aren't any really good biomarkers that accurately predict risk of comorbid disease. So the important principles, one is, where is the opportunity? There are a lot of comorbidities. Which do we target? You asked earlier on, Johan, where should we look? But also we might want to think about which are the most important ones to focus our attention. But firstly, better diagnostic biomarkers would help the whole field. For example, in psoriatic arthritis, the diagnosis of that is very difficult. If you're developing a biomarker, a prognostic biomarker, and the outcome is so difficult to measure, that's challenging. But as also Sapphire has said, the opportunity to intervene early and potentially prevent and modify that disease trajectory, although that is still to be proven. As Christian said, the term biomarker is, I like this FDA definition because it's very, very inclusive, but it's important to remember that if your biomarker is mechanistically related between your exposure and outcome, it's going to be a better biomarker than that that's further away from the causal pathway. And that's why the effort and discovery around molecular mechanisms is so important. The other thing just to sort out is terminology. It's certainly an area that I sometimes get confused, but a comorbidity is a distinct entity related to your index disease, psoriasis here, whereas multimorbidity is a coexistence of any two or more chronic conditions. And of course, multimorbidity is very common in the general population, and this is also important when you're thinking about biomarkers in the context of psoriasis. So this study took around 4 million patients in the UK health records and defined multimorbidity as ever occurrence of two or more conditions over the lifetime or period of documentation in the same individual. And that's important when you're thinking about comorbidity development and therefore biomarker development, because of course the biological mechanisms driving that comorbidity may occur decades before the disease manifests. So the point I wanted to make here was that there's a lot of prevalent comorbidities that are in psoriasis that are obviously very common in the general population. Whereas, and in the same paper, if you look at those morbidities that occurred more often than expected by chance, those nodes relate, the blobs relate to the condition and the size proportional to the prevalence, and the thickness of the lines, the importance of the correlate. So I think this is a sort of principle that's important when you're thinking about your biomarker. Where are you going to deploy it? What's the target population? So where are we now? Well, as Christian's mentioned, weight is a perfect clinical biomarker. I mean, it's incredibly predictive, possibly difficult to address, but it is a very strong risk factor, not only for development of psoriasis, but also for the progression to psoriatic arthritis and, of course, cardiometabolic disease. And these biomarkers are integrated into routine clinical care already, with risk thresholds for intervention in the general population. So the SCVD is used in the States. We have the Q-risk in the UK. And there has been an argument to say these need to be validated in the psoriasis population, but and one such validation study in the CPRD suggests that actually it doesn't perform badly, particularly if you're just looking at psoriasis. The AUC there is 0.815 with a very high negative predictive value, although the problem is that the positive predictive value is low. And as illustrated by Alison earlier on, of course, cardiovascular disease outcomes are higher than that. They're there, but they're not that common, and that's probably we need very precise biomarkers to be strongly predictive. And in fact, we scoped the research landscape, and although this is a couple of years old now, it hasn't substantially changed in terms of the principles, in that there's a whole mess of biomarkers out there, but most of them are in the discovery phase and haven't been validated. And we picked out some candidates, and the detail probably isn't important, except to say that it's interesting that some of them seem to go across multiple groups of what you might call disease progression, so severity, arthritis, and diabetes. But these are the challenges in this area generally. So on a positive note, there are these very large, ongoing, collaborative, data-driven research efforts to identify clinically useful biomarkers. I'm just going to talk about a few things that I'm sort of involved in with our group. So first of all, if you think about comorbidity, it does occur in the more severe population. And so could we better identify the more severe population? That would be important for trials if you were going to intervene early and see if you could progress. And it also might be important for developing biomarkers. And so looking across multiple biobanks, there's evidence that individuals with a higher genetic liability for psoriasis are also more likely to develop severe disease. So as Satvir showed, this is being integrated and looked at across 4 population-based biobanks, so that the next stage will be, if you can integrate these, do you get a better prediction of more severe disease? And also, how does that relate then to subsequent development of comorbidities? It may not be any better as an integrated multimodal biomarker. Psoriatic arthritis, I think this is something that's really worth going after, but it's challenging. This review is quite nice because it does summarize essentially. It's a bit old, but it does summarize more or less the state of play, in that we know that there are clinical risk predictors, subphenotypes of psoriasis, a family history, more severe disease, obesity, and then there are more serum biomarkers published now, but essentially they're all still at the discovery phase, not in clinical practice. But in reference to the outcome and this idea of subclinical musculoskeletal inflammation, there's been a change in the terminology to try and make this more precise, and also then when you're sorting out all of the different cohorts, which Hippocrates are trying to do, and aligning and curating them, you can then determine your cohort, how the PSA was diagnosed, was there a prodrome, and try and get some commonality in order to really interrogate then the underlying, the collected molecular data. And as part of this, there's this massive effort across Europe to collect 25,000 patients with psoriasis who may then develop to try and pick up instant PSA, associated then with collecting DNA from this cohort as well. So it's a potentially really powerful tool. And we'll pick up, Nick, as you said, these patient-reported outcomes of change in symptoms and patient-reported outcomes over repeated times. And finally, cardiovascular disease. So this is, I'm nearly finished, but there is this prevailing view that psoriasis is a systemic disease and that is the inflammation that's driving the cardiovascular risk. But as Christian alluded to, of course, there are lots of confounding factors that make unpicking this relationship very, very challenging, because the risk factors that we see in the psoriasis population are also risk factors for cardiovascular disease. And so, as you will all be familiar, this technique, Mendelian randomization, where essentially the genetics are used as an instrumental variable that's not affected by that confounding those confounding factors, to try and get around this problem. And so it does have its own challenges, Mendelian randomization, and Sarah is a much better expert than I am. But nevertheless, it can help to try and unpick the causal direction of effect. And Ravi, who's just finished doing his PhD with our group, has looked at this very systematically to try and unpick this relationship. And this is just about to be published. But in short, what he found is that the genetic predictors of both coronary artery disease and stroke increase risk of psoriasis, but not the other way around. And in fact, those genetic predictors are those shared by coronary artery disease and stroke, suggesting that the biological pathways influencing cardiovascular disease contribute to psoriasis risk. And in the context of biomarkers, it may well be that we've been looking in the wrong place for those biomarkers. So just to summarize, we do have risk prediction tools that we're already using in the general population. Should we be using them, and could we be using them just more systematically in our psoriasis population? I do think that we need to understand mechanisms first and then develop the biomarker afterwards. Otherwise, you're fishing looking for a needle in a haystack. And with that in mind, perhaps we need to think about better stratifying the population at risk for those mechanistic studies. But, I'm optimistic. The opportunity is great. And just to say thank you to everyone I work with. Thank you.